Thank you guys so much for being here. Thanks for coming in. We got some cool stuff going on that we need to cover. I actually wanted to cover some stuff that we didn't get to last time. So there's some cool science news that's happening in the world. So we need to go over that. But I hope your day's going good. Thanks for being here. Um, yeah, hope we can have some fun, go over some science, figure some stuff out together, and answer some questions, if anyone has them. So, all right. First order of news. First order. We got NASA. NASA's plan for a nuclear reactor on the moon could be a lunar land grab. It's kind of an interesting idea. Here we go. NASA could soon be going nuclear on the moon. The space agency's acting director, Sean Duffy, has issued a directive to expedite building a nuclear reactor on the lunar surface. Duffy, a former Fox News host, is also the head of the U.S. Department of Transportation, and he took over leadership of NASA in July. So he's both things, the U.S. Department of Transportation and the head of NASA, after the Trump administration pulled back its nomination of private astronaut and businessperson Jared Isaacman. I'm a little confused. So he's both of these at the same time? It's okay. Whatever. I guess I'm not surprised. The directive, first reported by Politico, would accelerate NASA's long simmering and to date largely fruitless efforts to develop nuclear reactors to support space science and exploration. I guess that makes sense. He is from the real world, which is wild, man. So many of these Trump administration people are from reality television. I guess I'm not surprised, right? I guess like a breeder reactor or something would be a way that we could get energy to the moon, and then we could use it as a little launch pad, maybe going into Mars or something like that. The space agency pursued projects over the years, mostly recently in 2022, when it awarded 3 $5 million contracts to companies to craft designs for small space-ready reactors meant for lunar operations in the mid-2030s. Inspired in part by a space policy directive issued by President Trump during his first term, those reactors were intended to produce 40 kilowatts of power, enough to sustain a small office building and to weigh less than 6 metric tons. Duffy's directive is more ambitious. It calls for NASA to solicit proposals for reactors that would yield at least 100 kilowatts. Okay, so more than 2.5 times, and ready to launch by 2029. The space agency is tasked with appointing an official to oversee the effort within 30 days. Interesting. Let's see, chat real quick. Fasting is the problem with fasting is just the ability to get enough protein and stuff in your diet. So basically, you can only assimilate about 40 grams of protein per meal. So if you did like one meal a day, you're basically going to be defecating a lot of the protein as opposed to assimilating and making it protein muscles. Yeah, I mean, it's I don't understand. Oh, thank you. I just think there's probably more qualified people. Like not putting it against them or anything, but it's just kind of weird. I feel like it's a little odd to nominate such uh unserious people. He just doesn't know, but he's not serious. I guess I don't. Thanks, Shy. I appreciate you saying that, man. That you got me. That was it. You're right. Lunar nights are very long, 2 Earth weeks, and previously cold, making nuclear power desirable for surface operations. But according to the directive, the greater impetus for the fast track plan is a burgeoning partnership between China and Russia to build a nuclear powered outpost near the moon's south pole by the mid 2030s. The sun never crests above the horizon there, leaving some craters in permanent shadow and valuable deposits of water ice lacing through their eternally dark floors. Despite its cryogenic chill, its lunar region is hotly contested, with NASA's Artemis program also targeting crewed landings there. I wonder if that's why they want to. Are they trying to go there to like cool the reactors down, or are they just saying that there's not going to be a place for solar panels? Um, Besides providing abundant electricity for surface operations, a nuclear reactor on the moon would also allow for a strategic lunar land grab. Ownership of otherworldly territory is prohibited, according to United Operations Outer Space Treaty, but the treaty also obliges spacefaring powers to exercise, quote, due regard in their activities, meaning that they should not encroach on or interfere with sensitive infrastructure built by others. So that is interesting. How would they regulate that? My research focused, I'm a microbiology PhD. I used nanotechnology techniques in order to research DNA delivery to algae chloroplasts. And I do genome engineering for yeast right now as a host development. So, yeah, I'm a research scientist. A nuclear reactor placed on the lunar surface, therefore, could allow the declaration of what Duffy's directive calls a keep out zone. Although the Trump administration's acceleration of NASA's nuclear power efforts may be welcomed by many space exploration advocates, it comes alongside other proposals from the White House that seek to radically reshape the space agency that would be a cross purpose for the new directive. This includes plans for an extraordinarily deep cut to the NASA's science programs, as well as an active and ongoing culling of the space agency's workforce. The president's budget request for the fiscal 2026 year notably zeroes out funding for a joint program between NASA and the DoD to develop nuclear rocketry. Why would they do that if they're investing um, into that? That doesn't make any sense. I'm very confused. The nuclear rockets seem to be the next thing. Oh, thank you. Thank you. Um, Interesting. All right, guys. Well, that's the NASA space exploration idea that is ongoing. I think, like in general, I think that it is good to have a space. Um, how much have you researched our politics? What do you mean? What do you mean by that? Hey, Puff Pass, how's it going, man? Uh, I'm gonna see if I can make you a mod. I don't know how to do that exactly. Let me see. Let's see here. Thoughts on the DC National Guard deployment? I mean, it's insane. I mean, he's just militarizing everything that he can in order to. It's like just a naked power grab. So it's unbelievably unconstitutional. Um, I mean, it's the Constitution's built on federalism, that the states and territories have their own ability to regulate themselves, and this is just a complete over. Uh, it's just a blatant unconstitutional power grab. Um, Kind of what you've already been saying, but like, do you get deep into politics? Yeah, of course. I mean, science and politics are intertwined. There is no real separation. So yeah, we talk about politics all the time. Yeah, he's welcome. It's all right. You can ask any. This is a welcome space, man. You can ask questions. It's all good. I appreciate it. But yeah, you can ask whatever you want. Um, Let me see if I can try to find moderator. Yeah, you're welcome, Tony. It's all good, man. It's all good. I'm trying to find chat moderator. More chat settings moderators. There they are. Oh, okay. I'll figure it out. I'll figure it out in a second. Do I believe in free will or determinism? I think it's tough, man. I think that there's some new research to show that there's people that you can actually re um, you can respond to things before you actually gets the central nervous system. So there is like some peripheral kind of action that happens. So I want to say that we're, biologically, we're probably a little bit more determinism, but I do feel like people are free in order to make their own choices. I mean, personally, I know that I'm a different person every year between like 18 and now. I've been constantly improving, constantly uh, im- changing. And so I can kind of respect that. Does God want us worshiping guns, wars, soldiers, flags, and militarism as many people are doing? I don't know what God is doing because I don't really believe in a God. So I don't think that it's patriotic in order to worship those things. That's what you mean. It's a breath of fresh air for some real intellectuals with all the stuff we were pummeled in. Thanks for saying that, Tony. Yeah, I'm trying to make a space where people are allowed to discuss uh, and you're welcome to ask what you want, man. You don't have to. We can build out a community that will be focused on compassion and learning and improving ourselves, just like science community is. And I say it a lot, but when I first joined my first science lab, it was a huge moment in my life that those people like coming together to make positive change and trying to make the world a better place really inspired me. So I would love for this stream to do that, in my account and this channel to do that. Can we be friends? I'll support you. Okay, I think that you, Nancy, I'm pretty sure you are a bot and block you. Let's mute you the entire live and we'll see what happens. How long were you in school for? I'm in San Diego. So I went to community college for 2 years, and then I transferred to UC San Diego and that was like 5 years in total. And then I worked at a biotechnology uh, company for 5 years, and then I went to grad school, and I was in grad school for 7 years. So in total, I guess I've been in school for like 13 years, grad and post grad. Um, so yeah. What kind of science do I do? I basically got my PhD in microbiology using nanotechnology techniques, and I delivered DNA to algae chloroplasts with single-walled carbon nanotubes for the first time. So we imaged the DNA with the dye, and it got into the chloroplasts. And the work that I do right now is with molecular biology. Basically, in the last 16 years, I've been doing molecular biology and genetics. So that's kind of like where a lot of my expertise lies. And yeah, I do basically change genomes of yeast in order to make them make proteins better. Nanotechnology is any particle. So there's nanoparticles that are less than 100 nanometers. So that's anything. There's a lot of different types of nanomaterials. But when we functionalize them and we change them to do specific things, now they become nanotechnology. And that's basically, yeah, that's what it is. So there's a lot of different types of nanomaterials, lots of different types of nanotechnology. And something I learned really shockingly is that we've actually known about nanomaterials and nanotechnology for a really long time. Uh, the microplastics in our bodies, that's a really interesting question. I think that there's enzymes that bacteria can do that can make like nylonase and other PETAs and stuff like that, where you can actually break down plastics. So, um, yeah, I think that that's probably the best way because plastics are nonpolar. So it'd be really hard. You can't just like pull them out with a magnet or something. You have to basically have something go through your body and kind of chew them away. So either like have an immune system that is used in order to fight those off, or you have like a bacteria that can do it. A Christian atheist. So basically I was born into a Christian family, and my mom and the values that she taught me were, to love one another, love thy neighbor, be nice to one another, use compassion, take care of the poor, the meek, all that kind of stuff. And, I eventually fell out of religion and I don't believe in God anymore, but I still have those morals. So the Christian atheism is a thing. There's a Wikipedia page on it. And it's kind of like how I explain my morals. I think that a lot of people can get to that position without, calling themselves a Christian atheist, but I feel like that is a easy way for a Christian, which is the majority of people that live in the United States, to kind of just understand where I'm coming from. So that's why I like to couch my morals in that. Go actually play with someone else's page, anyone's. Find out what you love and express it your way. Okay, cool. What do you think of vegan plant-based diet and what's the key to prevent disease? So I think that like a big thing people get wrong is that they have to eat meat on every meal, and I don't think that that's correct at all. Actually, I think that you can, eat meat sparingly and still get your protein in. So you still want to get 200 grams of protein every day. I weigh 180, so I'm getting like 200 grams of protein a day because I weight lift and I do jujitsu. So I think that's very, very important. Um, the key to preventing reduced disease, is just not being morbidly obese. I think that's the biggest thing, is just consuming less food. So in our society, we have a lot of tasty food constantly available. A lot of people are stressed out from being exploited at work. They don't feel like and they're basically stress eating. So, and that's kind of like where a lot of the disease comes from, is that people will get uh, you know, diseases related to eating too much. So that's the number one way that people can prevent disease. I don't think that like processed foods or preservatives are inherently unhealthy or filled with poison. Name one thing you've actually accomplished besides taking millions in government grants. Hmm. I mean, I've worked on algae biofuels, trying to create a renewable and sustainable fuel production. I've created plants that take up less pesticides than their organic counterparts. I've pushed into a new interdisciplinary space for the first time, and I've created a new algae nanotechnology space that was really never explored, and I have a patent in that. So that's like a something that I've contributed, and I've created yeast strains that have increased protein production significantly for pharmaceuticals that people use. So, yep. That. No paranormal or spiritual experiences, really. No. Genetics facts. There's 23,000 genes that you have in your genome, but about 100,000 different types of proteins. So I think it's a really interesting way of like kind of thinking about all the different ways that those genes are turned on, spliced together in new and interesting ways. Blue pigs. I don't know blue pigs. RFK Jr. is a bad person. He causes people to die. He has directly been linked to island. He's gone to several nations and caused vaccine hesitancy, and that caused he's caused like measles outbreaks in these island nations after he went and caused vaccine hesitancy there. I think it was like Guam. I'm not 100% sure, but it's awful. He's an awful person, and he's gonna cause people to die. Millions in grants is an accomplishment. No, they are very hard to get. uh, oh, thank you, Rocky Rush. Not yet, not yet. Um, yeah, yeah, I was actually very fortunate. I actually got, for my graduate fellowship, I got two prestigious grants, fellowships when to pay for my schooling, and most people only get one. So it was actually very rare to get two, and I was very, very grateful for that. And I got my PhD funded by the Department of Defense, the Army Research Office, where they were interested in the play between nanotechnology and microbiology. Uh, we worked together. That's how I met my wife. I only have one. So that's, yeah. Oh, thanks, Astral. Thank you. There are pigs that are turning blue from an invasive squirrel poison. Oh, I don't know about that. Let's see. Let's check that out. Blue pigs, California squirrel. How many people have died of the measles? I don't know. But when you make people vaccine hesitant and you cause people to die because of that, then it's more than should have died. Is now a bad time to get a PhD? Man, that's a really interesting question. I think that so when I got a PhD, I literally had nobody recommend me get a PhD. Even my friends with PhDs were like, "Don't get a PhD. It's not a good idea." And I did it anyway. So, the thing is that you need to have that tenacity and that grit to say, it doesn't matter what you think, I'm gonna get this thing and this is what I wanna do. And you really have to have that determination in order to kind of like make it through all the downsides and the mental health struggles of getting a PhD because it is very arduous. How many people have died of the measles? None, that's how many? None. Okay, interesting. Okay. I'm pretty sure people have died of the measles. How else would you know about them if you people haven't died, if it's not an infectious disease? Has autism always been as prevalent in society as it is now? I think that it's more common because we actually have people looking for it. I think that as we have more ways of studying things, we're going to have more detection and we're going to have more people found. It's kind of like the cancers that are coming out now that there's more and more cancer because we understand the molecular biology and the genetics more. So we know the abnormalities more. Something wasn't considered a cancer before, and now it is. So that's what's happening, in my opinion. I'm not a medical doctor, so this is my non-medical doctor opinion. How many people have died from vaccines? Guillain-Barré's disease is extremely rare. Vaccines are safe and effective, and you should get all of them. So, uh, let's see. What do you think of DNA sites with 23andMe and Ancestry? Man, I think it's a really interesting idea. I love the idea of people being more connected to their genetics. Hello. Hi, Jaxie. I love the idea of and I actually think that like sequencing DNA is actually going to be a bigger part of our healthcare system going forward. So, like for instance, I think that when you go to the hospital in the future, you're going to have your gut microbiome kind of constantly sequenced in order to see how that population is doing and thriving, or if it's not, or if there's some type of like staph infection kind of taking over. And it would actually really help in preventing disease and creating alerts for things like MRSA and stuff like that from stopping that from happening. So I think that it's going to be a bigger part in the future. Does the earth naturally provide everything we need to heal ourselves? No, no, definitely not. I mean, that's why like science has been the one that's actually advanced humans' life expectancy past 30. I mean, think about the 200,000 years that humans have been around. It's only in the last 100 years that our life expectancy has really gone past the age of 30. And there's a reason for that. People used to eat all organic foods with no preservatives and, no genetically modified foods. And they would still die at 30. So, can you recommend someone to learn about the COVID testing? Do you mean like the tests that happen, or do you like what type of tests? So basically, there's an antibody that is produced in your body, and that is um, binds to an antigen on the test, and then that flares on the little COVID strip test. There's also PCR, and PCR is another way that you can amplify the parts of the virus, and you can see what if those virus parts are still there. CRISPR didn't come from the earth, it came from scientists. Almost, Sherry. I mean, that the technology, you're right, it came from scientists, but CRISPR is actually a bacterial defense mechanism. So, CRISPR-Cas9 is a way that bacteria used to stave off infections from other viruses or bacteria. Basically, it's kind of like a hit list of the worst sequences that it's seen, and when it goes out, it finds those sequences and it takes it out, kind of like a targeted shot. I always wonder if we should have extended our lifespans, or we must. Hey, man, I'm 38, so I would rather not be dead right now. Yeah, anyone's welcome to join Sigma. Yeah, I'm a research scientist, not a medical doctor, but whatever you want to talk about, it's all good. Yeah, Aiden, if you want to queue up, you can totally do that. We'll usually do like a 10-minute thing. So if you want to join, we'll just do 10-minute timers, and then the other person can come in and we can kind of go around and around. That's kind of just how it's nice to let other people kind of join the discussion. Simaga, how's it going?
Yeah, hello. Thanks for having me
Hi.
there.
Yeah, of course, man.
Hey, it's fantastic that you're educating people and offer your expertise.
Oh, thanks. I appreciate that.
I'm still a little confused about how they did the testing during the COVID
Okay,
years.
sure.
Especially, like I understand the basic principle of PCR tests, but I thought I would understand like how diagnostic tests work and how they are statistically um, well defined and evaluated. Because what confuses me is that the accuracy of the test was just too low, given the prevalence of the disease.
Okay,
I mean,
so
you probably know, like I don't know if you're not a medical doctor, maybe you don't know, but like there are like basically two numbers that define the accuracy of a diagnostic test. Let's say a pregnancy test, right? So you would have one number that determines the false positives and another one that determines the false negatives. And if you use those numbers combined with the prevalence, you can determine the actual numbers of false positives and false negatives. And if you do the math for realistic numbers, the you're getting, for example, for Australia, you get about 50% or more false positives, and I think that's a problem.
okay.
And that's just that's regular simple math. There's nothing um, controversial about it. I checked this with other doctors
Yeah, so
who
what you're talking
confirmed.
about, so there's like the epidemiologist, and who basically does the forensics of the pandemic, and then there's the like the mechanistic physical testing of like whether or not people are positive or negative. So you can have RNA detected by the swab, so they're detecting the protein through like the swab, right? You can have it detected by the RNA in again, so you can have RNA test, you can have a protein test, or you can have your blood taken and you could check for antibodies there. So those are like the three ways that you can have that. Like, are you SARS-CoV-2 positive or negative?
Yeah,
On a
I
grounder
understand that.
scale, on a bigger scale, you have kind of like expected deaths. So you have the people that are expected to die every year, and whether or not you have more or less of that. And then there's also kind of like the genetic forensics of epidemiology. So basically, we can look at a population and see what the expected mutation rate is of an RNA virus, and we can look at the rate of viral genome recombinations that happen, and we can actually kind of
Yeah.
like surmise what's going on. That with a population rate of like it's actually sewage water testing. So sewage water, as people defecate, there's like RNA in there, and RNA is detected, and basically biobot.io was like the main website that everyone used, and it basically detected the rate of SARS-CoV-2 infected at the city in the population level. So those are kind of like the ways that everything is kind of all come together. There's a couple of different websites,
Yeah.
covariant.org, that people use in order to kind of track it all.
Well, I would like to focus on two tests, like the PCR test and the rat tests that were used in Australia, especially, and then I guess in most countries.
Okay,
And
well, all
they
those
made,
tests are used everywhere, just letting you know. But I mean, so
yeah.
it's kind of like all of them together kind of give a bigger picture, because you're not going to get a population level understanding of the rate of the virus if you're just looking at positive or negative PCR tests.
Well, what confuses me is like let's forget the that it's COVID. Let's say it's a
Okay.
pregnancy test. So
Okay.
any diagnostic test follows a specific formula, like equation, where you can calculate the false positives.
Uh-huh.
And if you put in numbers like specificity, like 99%, and sensitivity 99%, and prevalence like say 1%. So which are like on the positive, like these are very like optimistic numbers, like the practical
No, that's
numbers
way
if you
too
look at
high.
it.
That's way too, like on a PCR test, that doesn't happen.
Yeah,
So basically
I
what you
know.
do is you have like,
I'm generous.
but you have like controls that you run. So specifically a PCR test, you have like controls that you would run in like without a template. So it's called an no template control, an NTC,
Yeah,
just to see if you
I
have
know, but
any.
I talk to people who run the tests. Actually, I know the details. What
Okay.
confuses me is the math behind, like because as you say, the numbers are actually too high. In practice, they are like especially the red tests are lower, and but I'm assuming like maybe the highest potential specificity. So it's more like 95 or lower. But if it's 99% and you run the numbers like for like a low prevalency, prevalence.
Mhm.
That's the problem. That just produces, a relatively rare disease with, has the problem that the false positives are very high, even if the specificity seems high. You would need 99.9 and maybe another 9 to
Mhm.
actually get something useful. And I mean, that's just the math
No, I mean,
based
so
on,
the epidemiology is actually that it was way, way, way more prevalent, so that there was a way higher population and community spread than people understand or realize, and that's actually the way research is happening right now. Is that people are curious about how there was so much asymptomatic spread in the population. So if anything, like we think that there was the numbers of asymptomatic
well,
spread is actually
I
way
know the
higher.
numbers. Sorry to interrupt you, but in Australia, we never reached the maximum was we didn't even reach uh, what is it, 250,000, which is sort of the expected false positive rate. Which is 1% of the population. Like we
Oh
never
yeah,
even
that's
got
no,
to
it's way more
1%
than
of the
that. Yeah, it's
population in
way more
terms
than
of
that.
case.
So again, I think you're misunderstanding. Like SARS-CoV-2 and COVID-19 are two different things. So SARS-CoV-2 is the virus that causes COVID-19. So you can be just like HIV and AIDS. You can be HIV positive without developing AIDS. You can be SARS-CoV-2 positive without developing COVID-19. And people get them mixed up a lot because colloquially and culturally we kind of interchange them, like, oh, I got a COVID-19 vaccine, but it's actually a SARS-CoV-2 vaccine.
Yeah.
And what,
Okay.
so
Understand.
people, it's called an asymptomatic spread, where you'll have a viral load that is big enough in order to spread it to one another, but it won't be big enough in order to cause COVID-19.
Yeah. Well, I guess what I'm saying is like if the prevalence is low enough, you would automatically create enough false positives to make it look larger. So in
Mhm.
case the prevalence was actually fairly low, then the test is just not suitable. But that's not even controversial, I guess, and that just confused me. But what
Mhm.
confused me even more is that there isn't really a gold standard. I mean, the gold standard was officially the PCR test
Yeah.
itself.
Yeah, it's very
So
good.
how can you even measure the accuracy of a test if the test itself is the gold standard?
Easily, because you can actually detect how accurate it is in a mutant library that's similar to the SARS-CoV-2 PCR like primers, and you would basically see like how much false priming there is. So basically, the amplicon is created during the PCR reaction, and that exact amplicon needs to be the exact same size. And if it's priming and the primers are landing on DNA sequences that they shouldn't be landing on, basically at the end of your qPCR, you have a melt curve. So a melt curve says, okay, let's say a DNA piece is 20 nucleotides long. That should be associated with a certain amount of energy in order to melt those DNA strands and that amplicon into separate pieces. But if they're more than that and there was false priming and the amplicons are longer, then well, it would require more energy in order for that melting to happen. And you basically, after
Yeah.
every qPCR, you have a melt curve, and you can check that. Like it's standard.
Okay, but then why were so many heating cycles used? Which are known to cause so many false positives.
No,
Like
it's the opposite. It's like
also they were not really published.
no, it's standard. It's because it
Like
gives the
40?
same
I've
amount
never
of
heard that
amplification,
before.
and it actually gives it a chance in order to actually be provable. Because if you have like if you're actually doing 30 cycles of a qPCR and you get no amplification from that, no template control, and all your melt curves are exactly right, and that's a really accurate test, and you can actually tell, and how many cycles it was, and you can discern basically how much it is. And honestly, because qPCR is so good at detecting like 1 in a million particles, you actually kind of need them to have a chance to find that like single amplicon and get that detection method. So if you think
But
about
that
it,
is
it's
also
like,
the problem.
hmm?
That's the problem. The
No,
false positives.
no,
It's too
no, because
sensitive.
the false positive would come up in the no template control. The false positive would, again, so if it was a false positive, then it's mispriming, and the amplicon isn't going to be the same size that you're expecting. It's going to be way bigger, way smaller, and you can tell that with the melt curve.
Yeah, but I mean, they were looking for the spike protein markers, right?
Mm-hmm.
And they. I mean, even if you pick up a dead virus or just a part of it, it would be a positive result, right?
It's going to specifically go towards that spike protein. I think it's actually, and then on top of that, you actually they actually amplify off the capsid too. So it's actually 2 parts of the genome that it amplifies at the same time. Because
Yeah, but
if you
is it
do
correct
only the
what I'm
spike
saying?
protein, you know what the problem would be? If
Say it
you
again.
just amplify the spike protein, do you know what the problem would be?
No.
You would get people that have the vaccine, right? The vaccine would trip that. So you actually have to
Yeah.
amplify
Okay.
off of the capsid, which is the structure that is only there for the full virus. So if both of those are positive, then
Yeah.
you
I
have
understand that. Yeah.
an actual
I
infection.
mean, that's correct. But it worries me though that like people were like shipped to the north of Australia because they were like positive on a PCR test,
Mm-hmm.
and they could have been because of a dead virus on them,
Mm-hmm.
which they have plenty of. I mean, all of us have dead viruses on us.
Not with
And
that specific sequence, no. I mean, that's, again, you can make these primers extremely specific, extremely specific.
yeah, but wouldn't there like that if there was a disease with viruses around, wouldn't we be surrounded with viruses of that kind anyway? And also
coronaviruses,
many
yeah,
dead ones?
absolutely. But coronaviruses with these specific sequences and that specific length and giving an amplicon that gives that exact amplicon length, no. I mean, that's why
Yeah,
it's
but
unique and
having
that's why it's
now I
cool.
understand, but I mean, just because we have like evidence for some virus components in our bodies,
Yeah.
that doesn't make us like a case. That was confusing for me because the definition of a COVID case was changed, and it became the test result, which is not reliable as it looks to me.
It is.
Like it's
It
not
is.
necessarily a real case.
It is
It's
a real
like a
case.
case.
It
I
is
mean, a real
because,
case in the sense that there was a virus particle found,
again,
but
a
not.
virus particle, you might get You might get a capsid from a similar coronavirus because the coronavirus is going to maybe have the outer casing that is similar, but you're not going to get the exact capsid protein and the spike protein in the same place unless it's SARS-CoV-2. And if you get just the spike protein without the I'm saying
Yeah, no,
protein, but
I
I'm talking
understand
about the
that.
RNA that yields the protein, obviously, right? Because
Let me rephrase.
you
I
do
think you
a
misunderstand
okay,
what I mean. Um, uh, do we have flu viruses on us right now?
yeah.
Like even if you're healthy?
Coronaviruses are everywhere.
Yeah, or any viruses, really, like flu viruses, not
Sure.
just corona, right?
Hmm.
So if I would test with for that kind of a regular flu virus of some particular type, I would find one on you and on me, right?
Yeah. What would happen with those qPCR tests?
Yeah. So it would be cases of flus,
Okay. And how would we
and
differentiate those flus?
yeah, that's what I'm saying. Like if you're testing for whatever, you will find it because we have it.
But what would you do in order to differentiate those? How would you tell
Differentiate
them difference?
from well, there doesn't need to be a difference. Like the it doesn't matter what you're looking for, you'll find it.
Okay.
So we have, if you have a particular, I mean, the initial sequence was just a number basically. It's a DNA sequence, right? Or some
Okay.
encoded protein sequence that basically can be defined through numbers, and that number sequence defines the virus or the virus particle. So that could be found like all in any of us at all times. So how does that make a useful diagnostic tool?
Because the viruses have different RNA genome sequences that we can amplify specifically.
Yeah, no, I don't doubt that it finds let's say the COVID virus or some flu virus you're looking for,
Okay.
but it doesn't indicate that this is a problem, right?
I mean, you're right in the fact that virologists actually think it's really interesting that SARS-CoV-2 is only the biggest thing that differentiates it from other coronaviruses is a plus 10% binding affinity to ACE2. That's it. So we actually got
Yeah,
super, super lucky that SARS-CoV-2 wasn't a super bad virus. It was actually really easy, and coronaviruses are really easy to deal with. We know how to, use PPE for them. We have vaccines that for them. We actually got extremely lucky with COVID-19 pandemic that it was
but
just a
how could
coronavirus.
we? Do you know? I mean, how was the disease actually diagnosed? I mean, I
Mhm.
can't believe it's just like a test that can basically find anything, just like its inventor said. Like it's just amplifying stuff, uh, and there are no specific enough symptoms. There's no, it's not like you're probing for like a heart inflammation and you take a biopsy, which could be the gold standard. Which and let's say a blood test is like um, is like the proxy for that. I mean, I would understand that,
It's just
but
basically we saw a bunch of people in China that were developing similar symptoms. They did some viral tests. They found that the SARS-CoV-2 where it was present in all of them, and then they did the genetic tracing, and basically they found the link to all these people. In the center was this wet market, and then they did sampling there, and they found raccoon dog samples with SARS-CoV-2 samples in it at the same place at these stalls. And then they did genetic testing at the wet market, and they found two distinct SARS-CoV-2 strains, 1 week apart, and that
I mean that's
never really.
correlation.
No, it's causation. Absolutely. When there's no
How?
explanation other than a natural zoonotic
What were
spread.
the? But the symptoms are very like generic. I mean, like that's my problem. I mean, if it was
Okay.
like some sort of red spots or some rare symptom, I would understand. But if it's
Hmm.
like a sneeze and a cough and whatever,
Okay.
it's
But if all those
it didn't
people
look.
have like the same SARS-CoV-2 viral load that's high, that's causing this problem, wouldn't that be a distinguishing characteristic that it is SARS-CoV-2 related?
I'm not. Does the test actually test for how high the load is? I mean,
Yeah,
would
absolutely.
that
With
be?
qPCR, you can absolutely, yeah.
but you don't really get that result, right? Like if I was when I was
No,
tested, it didn't tell me how high the load
you
was. It
can.
was
We
just
did.
either
There's
positive
tests.
or
There's
negative.
publications that do it. Basically, you do a 1 to 10 serial dilution, and you figure out, okay, so I'm going to take this much of RNA, and then I'm going to do a serial dilution in order to see
I
how
mean, I
much.
understand it can be done.
Yeah, I mean
I don't doubt
yeah,
that.
that's what
Yeah.
it is.
Yeah, but I mean, I understand that the PCR test alone, I mean, is already quite, a lot of work involved. There are like several people involved to do that, and it's not commonly done. This was actually, I mean, it's
PCR
a much more
tests?
accurate the PCR test, right? It's like relatively, I mean, takes a while. It's
No, it's super easy. I've done probably a million PCR
oh
reactions.
really?
Oh yeah, it's
Okay,
everyone's.
that's
In molecular
interesting. So
biology,
did you do
the
it
tool
alone?
the tool is literally like the hammer and nail of molecular biology. Everyone uses PCR. I did
Mm.
300 PCR reactions today.
But is it usually used for as a diagnostic tool, or is it?
Yeah,
I
absolutely.
mean,
The presence or absence of nucleotides in a place. Oh yeah, absolutely.
mhm.
Yeah, we use it
Yeah, but
constantly.
again, I mean, what made me a bit nervous was the fact that there was no gold standard. Like it looked like a circular reference. Like the gold standard was the test
No, like I said,
itself
there's
for the.
there's multiple. So again, there's
Yeah,
the
I mean,
PCR test that tests for the nucleotides. There's the serum that in the blood that looks for the antibodies. There's the protein that looks for the antigens, like the antigen tests. And then you
yeah, but
also had that
I'm
at the
not doubting.
population level of the biobot. You had and the wastewater,
Yeah.
the RNA and the wastewater. I mean, there's tons of stuff. We have full genome sequencing of it. It's not just PCR. It's the entire
Okay.
genome being sequenced. So it's we definitely
Here's
have
an
evidence.
example. What if, let's just hypothetically like assume that there is a new virus going around for
Yeah.
some reason,
Hmm.
and it shows up in all the people, but it doesn't actually do anything.
Okay.
So we would, with the PCR test, we would find that and other tests, right,
Hmm.
easily. But and then you know, but that's all. Like then maybe there are like some like common symptoms that are associated with a million other things.
Okay.
I mean, how would you be sure that that virus is responsible for those symptoms? You know, how would you diagnose the disease like that? This is actually caused by that virus and not
Um,
something else.
um, this is one of the biggest things in microbiology is that oh man, what is the process called? I'm dying. Oh man, what is it? Chat, help me out. It is the so
What?
basically, so in order to prove Koch's postulates, that's what it is. So basically, this is dude, you're getting to the main question of germ theory and microbiology. I mean, this is a great question. So Koch's postulates basically is that you take something that is diseased and then you see that thing that is diseased and then you isolate the microbe that's associated with it, and then you take it out and then you culture that microbe, okay? And then now you go to a third party, probably like a mammal or whatever it is. If we're humans, we're doing an animal testing, and then you have to
Mhm.
reintroduce that um, microbe to that mammal, and you have to have the exact same disease symptoms seen in that
Actually,
mammal than the
okay.
first one, and then you isolate it again, and then you test those two isolations, and then in this
Mhm.
case you would do some microbiology like or molecular biology, genetic testing. You look at the sequencing and all that kind of stuff, and if those two are the same, well, now you've isolated
Mhm.
the variable, right? You've isolated the
Yeah.
microbe causing the disease in this organism. You isolated
Mhm.
it and you say in this organism and you isolated it.
Wow, that's the first time I heard an explanation. Actually, that makes a lot of sense.
Yeah, it's
What's that called?
KOCH, Koch's postulates.
KOC, Koch's post. Oh,
Yeah.
okay. Okay.
Hmm. It's actually
Yeah.
it's really interesting that you bring this up, and it's actually one of the biggest problems in microbiology, in my opinion. I'm kind of like this new generation of microbiologists, right? That kind of I
Mhm.
don't know if we can. Koch's postulates actually says that we actually have to do this for all different organisms. In my opinion, I'm kind of like new wave in this thing where I actually kind of believe that we should be able to do Koch's postulates with molecular biology without actually isolating it, because it's actually hard to isolate and culture microbes. 99.8% of microbes that exist in the world have never been cultured in a lab. So how can you cause and associate it with a disease if you can't even culture it, right? So we should be able to do this with like molecular biology, knocking out the genes or something inside of that organism in the
Why
organism.
were they not?
Hmm?
Why? I mean, it cannot be done in principle, because, or is it like so difficult, or is it like because there's so many?
Well, so basically, in order to culture something, you have to isolate it. And microbes live in a community. So microbes live in a community where some are taking my waste, and because normally some are making waste, and then that waste makes food for someone else, and then that gives off a signal that another microbe says, hey, let's grow. And then once that grows, then another one's growing. It's kind of just this revolving community of waste transport, making sure it's not toxic for anyone, there's enough nutrients in the community, all that kind of stuff. And when you remove that, and you remove just a single species or a single strain, it makes it impossible for them to grow. So what we have to do as microbiologists that are trying to isolate organisms is that you have to recreate that. You have to recreate all of the nutrients. You have to recreate all the, it's called quorum sensing, which is when the microbes can sense one another and say, okay, let's grow. Let's do the protein thing or whatever. This is a huge problem. I mean, this is the reason actually why we didn't know that there were any microbes or viruses in ocean water until the late 1990s, is because we basically had to have like microscopy get up to the level that we could actually individually see individual cells and viral particles in ocean water. And we just couldn't see it. And we kind of knew that it was there. Why wouldn't it be there? But oh, thank you. I appreciate it. So it's there's
Yeah,
just so
that's
so
super
much that
interesting,
we're
actually.
learning about microbes, man. They're just everywhere.
Yeah, I see. That's a little bit like in physics, the uncertainty principle, where you basically distort the object when you measure it. So
Hmm.
you can't really isolate, like truly isolate these microbes without changing them too
Yeah, you
much.
have to supplement the media. You have to supplement their molecular mechanisms. You have to
Mm.
supplement everything that they're getting. And what we're learning now with next generation sequencing is basically we know that these microbes are in communities. There, we used to, our old thinking was like, okay, we're gonna have to study these things one at a time, or we'll study this strain and then study this strain and then study this. But now what we realized is that, man, these microbes are living in communities everywhere.
Mm.
They're living in communities in the marine water. They're living in communities in roots of plants. They're living in communities inside of our gut and on our skin. And basically, next generation sequencing has allowed us to take all the DNA from that entire community of microbes, smash it all up, and sequence all the genomes. And then through computer science, we can basically assemble the genomes back together. And then we can see the individual bacteria or microbe species. And then we can count how many there are. So we can, like, it's wild, man, the amount that we can actually know about microbial communities now.
Hey, so are you saying? Do you think like this has been done in the case of COVID, that they actually did
Yeah.
apply a Koch's principle?
Yeah, yeah, absolutely. You can. There's the early
Are
papers
there papers
in
on this?
late 2019, early 2020s, January 2020. I think the first like the isolation and the first sequencing of the genome came out. Dude, people knew about this in December of 2019. I mean, I obviously I was in a microbiology PhD program. I was kind of like watching this happen and reading the papers, going like, "Whoa, this is crazy! Wow, they're doing a really good job of like getting on top of this. This might not even get out." And then it was just like horror shows of like, "Oh no, we're not taking this seriously at all. Oh, this is awful." And then sure enough,
Okay, no, but I'm talking about basically isolating the viruses and
mm,
then transplanting them to other,
yeah, yeah,
humans, I guess.
not humans, but
Or,
that's an ethical concern, right? They would
yeah,
do they would
I don't
do
know how
mammals.
did they do it, and
Yeah,
trying
they
really
would do
to.
mammals, probably rodents or something, or I don't know, maybe since it's since it's lungs, I would maybe think it would be pigs. I'm not 100% sure. I don't do mammals for my research science,
So
but that's what I
so
would
so
think.
pigs could get COVID. Like I know
Mm,
that they tested fish in in China,
yeah.
uh, but yeah, okay. Um, yeah, that's hey you. This is fantastic information.
Oh, cool,
And
man.
yeah, it's really nice to have a life where you can actually have a sort of knowledge exchange.
Yeah, man. Yeah, dude. Yeah, all types of questions and everything are all okay here. I totally understand. It's I just want to give people an opportunity to ask a scientist like what, for all
Yeah,
the
I
stuff, and I
mean
love it.
you know the problem if the false positive still is a problem, but it
Mhm.
I understand you can't answer all these detailed
Yeah, it's
questions
just
here, but
it's basically you would test that by basically putting it in, so we can synthesize DNA in specific
yeah, I was
sequences.
just
So
yeah
you could
yeah
make different changes to the sequences, so that
and
if it was one or two base pairs off, would it bind here? If it's this and this, would it bind here? And you basically test that. You test a bunch of different primer sequences in order to see like which one is the most exact, which one has the least amount of false positives, and that's actually done a ton of work beforehand. So I've designed qPCR primers myself. There's like a whole equation that you do. You test like different quantities of DNA or RNA in this case, just to make
yeah
sure that, because if there's more DNA, then there's more chances in order to false bind. So then you're
yeah
testing that. You're testing different cycle lengths. You're testing different annealing temperatures. You're testing all these different things, and to make sure that you're basically getting only the specific uh,
yeah
nucleotides that you want. So
but
it's a lot
okay,
of like
I
molecular
understand.
biology that I've
Yeah
done, but it's kind of like it's hard to wrap your mind around if you've never done it all the time. But I assure you that there's everything
yeah
in our thing. And to be honest with you, when they did do that sequence, when they published it in January 2020, I looked, because I'm like, qPCR is kind of like my thing as a molecular biologist. I've done it probably for
mm,
15 years now, so kind of it's like one of the few techniques that I'm truly an expert in. So I actually did my own primer analysis on the WHO primers, and I was like, wow, these primers are really good. This is pretty cool. And then because Trump is an idiot and he decided we're not going to use the WHO primer set. The United States is going to do our own primers. So I was like, okay, let's test that. Let's test the new primer set. And I'm getting all of these like bad priming, like the equations and stuff are giving like way worse results. And I'm like, these are going to give like really bad results. Like, this is not a good idea. And then sure enough,
yeah
for months, man, guess what happened in the news after news? We're getting a bunch of false positives and false negatives. The qPCR test wasn't real. The results weren't trusted, and I'm just going crazy here, going like, this was so easy. It was so easily known. You literally could have just plugged it into any molecular biology program, and you could have seen the false priming that could have been done. It was just really bad priming set, but he was just abstinent about doing his own thing. He didn't believe that SARS-CoV-2 even existed, and all that nonsense, and
yeah
he caused the same problem that you're talking about.
Yeah, I mean, most of what you're saying, like, is happening before you arrive at the results. Like, you're talking about the process and how
Hmm.
it's done. But when I just look at the diagnostic key parameters, like
Hmm.
specificity and sensitivity, that's all included, right? Like, that's once they are published. I mean, if you sell a test, you sell it with the parameters, right?
Hmm.
Those numbers.
Yeah, but you have
And
controls. So you like in the thing, you have a positive and negative control. So you have a positive
no,
control that.
but people who use the test apply the final test. Like if you do a pregnancy test yourself,
Mhm.
you don't have a control. I mean, maybe you have actually on that the stripe. I guess there are like two stripes or something. I don't know. On there, like but I mean, with the rat tests, do you use a control there somehow?
Yeah, so I mean, these things are so dialed in, man. Like within like it's a 30 cycle test, and that at like cycle 17, 18, 19, you're going to see this amount of cycles, this many amplicons at this level. Like
Yeah,
you
but this
you
is
know it.
already captured by the numbers, no? I mean,
This is
if you
already
get a
captured.
99% specificity,
Hmm.
it that's in it, right? that's yet you don't have more than 99 or 95, right? Whatever it is, that's the number. And I'm just saying, like, if you use 99, which is much better than most tests are,
Mhm.
you have a problem if the prevalence is low. I mean, that's just a
I guess
known
I guess I'm saying it's better than
problem.
99% because it's literally you have controls and you would see if there was a false priming event, and you would basically see it come up way earlier in the amount of cycles, because the cycles indicate how many cycles it's gone in order to replicate in order for that priming event to happen. And if it's priming, like false priming, then it's going to be prevalent in the sample everywhere. So you're actually going to need less cycles in order to amplify the DNA in order to get to the same amplicon, like the
Yep. But
amplicon
again,
amount.
you're circling back to the before the published
No,
numbers.
no, this is
Like
during the
I'm
no, this is during it. So this is like you have tests within the qPCR for these positive tests that you're talking about in order to make sure that like this is exactly matching up with the
okay, but what do you calculate with that equation then? I mean, that's in all medical textbooks. You know, if you look up diagnostic tests, how do you calculate the positives and the false positives and false negatives? There's one equation, and that has the input parameters, those two numbers,
Yeah, so
and
you
then
you get a
the
priming,
output.
you get a primer specificity in the beginning. So before you get a primer specificity, you choose the exact primer set that you need that has the most specificity. And then now when you're actually on to actual samples, what you have is you have a bunch of diagnostic tests that are going concurrently with your actual testing sample. So you would have making sure that there's no false priming, there's no negative, there's no contamination within
Okay.
your reagents, that the primers are actually priming and giving the amplicon length that you expect. There's a ton of tests within the test to make sure that that's an actual result. And
So
you're
you're
doing
saying
all
in
these like
practice
three
it's
different
fine.
times too. It's all in triplicate to make sure that all of them line up. It's not just like a one-off thing, because sometimes like
Okay.
mistakes
Yeah.
happen. So it's called technical triplicates. So you would have each thing done 3 times, and then honestly a lot of that is
Okay.
done called biological triplicate too. So
Okay.
each
I want to
sample,
save your time. I understand your point here, but
okay.
what about the non-PCR tests, like the rat
Hmm.
tests, which were like probably
Yeah.
90% of all tests they're much cheaper and fast, and they were mainly used, what I've seen in at least
Yeah,
in
so
Australia.
once you get to like, this is why molecular biology is kind of like considered the gold standard, right? Because we have all these internal testing and all that kind of stuff. Once you get to antigen and antibody testing, now you can kind of get into some weird stuff, but it's still pretty good, positive or negative quick tests, because the real thing with the antigen tests and the antibody tests from the cheek swabs is just how quickly you can get it done. Thank you. So the idea is to really get a rapid test. The PCR test kind of takes a day to get back. That's kind of, you get
Yeah.
a lot more specificity. You get a lot more. You basically know whether or not it's
Okay.
there or not. But it's different
But
ideas.
you know,
Different.
in Australia, we didn't. I mean, if you're testing positive with the rat test, you don't even have to prove it. Actually, or you didn't have to do that at all. Like they were quite keen on getting positive results. It seems, and I mean, considering that the sensitivity and the specificity, especially of those tests, were much lower than the PCR, right?
Mhm.
At least not higher than 95, certainly not 99. I mean, just using that equation that is in every textbook gives you this, like, it just makes it not suitable for a prevalence of around 1%
You
or a
kind of
bit
just
higher
know,
even.
based off of like, so this is again, so you have not just like, you have different in epidemiology, you have different tests
Yeah,
that
but the
are kind
people
of like
don't have
all
that.
out there, and then they should all kind of like come into the center and
Now,
give a
I
similar.
think the argument you could make here is like that if you take it together with symptoms, with some, you use a Bayesian principle and you have some prior probabilities that you can take, into
Mhm.
account. Yeah. Like, if you have specific symptoms, if you have like red dots, and this is, an indicator of the disease, then it's much higher. Then you have a much higher, smaller false positive rate.
Mhm.
But if you don't, like if the test it was just taken by its own, and people can have symptoms or not, or the symptoms are very generic, I think this was just a little like too not scientific for me. Like as a thinking scientist, I was uncomfortable with this approach. I understand the PCR tests could be more accurate if you combine it with other things, or if you go to a doctor and they look at some symptoms. But if most of the population is just self-testing and self-reporting, and it's not surprising that you. I mean, we didn't even reach the expected false positives ever in Australia.
Yeah. Yeah, man. I mean, dude, this is why the testing, and you're right, where it's like, how do you know who's testing, who's reporting it? How do you know if they've done it correctly? Are they adding in the throat swab and the nasal swab at the same time? Are they doing? There's a lot of variability there, and this is why a lot of epidemiologists, they don't really. I mean, they don't. The numbers for the cotton swab stuff aren't really that important compared to the wastewater. Do you know what oh man, I think the problem is that biobot.io got shut down because of funding.
I know they were doing this. In Australia, they reported the results of the sewage measurements. But you know what? They also reported every day how many people died of COVID, right? Which was like in the order of 0 to 30, 50, 30 or something per day.
So you should, there's like expected deaths. Yeah. So you can look at biobot.io, and that gives us like a population level. So it's like,
yeah, that's what I did.
Okay.
I did that. Well, okay. I did do the calculation, and I mean, every day in Australia you have about 900 people dying, maybe 700,
Okay.
And then they're reporting the 30 who died with COVID, and you don't mention the like 670 other people, and even most of those are over 85. Mm-hmm. So I just didn't find that very like objective. That was my problem. Like, it was just all a bit too sensational, the whole thing. I mean, I understand maybe they want to, make people fear the disease and then take the vaccine more, but it was just working against them, I think. At least people who realize this, there's a lot of inconsistencies that didn't make sense, and maybe there is. I mean, I don't believe like you can really make a safe vaccine in a few years. Like, I mean, without a time machine.
For sure. I mean, it wasn't just a few years. I mean, it was used and developed since 1989, and it's incredibly safe, and it was very effective. It was actually the most effective vaccine that we've ever created. It was the efficacy of the original SARS-CoV-2 vaccine was 95%, and most flu vaccines are about 60 to 70%. So it was actually kind of like, it was going so well, and it was so high in the efficacy that it was actually immoral to stop it. Like, it was going through all the same stages. This is the huge misnomer that the vaccine didn't go through all the stages. It did. It went through all of the same 4 stages. It was just blazing through them because it was that good.
It just like this again, like it's a little bit difficult to see how you can say if something is effective if you can't safely say who is actually infected and who is not, and define the disease.
Well, those people.
But I have one last question.
Those people are being monitored, obviously, right? And they're giving like dosages, and they're being exposed, and they're being tested.
Yeah, but how would you even know you have exactly that disease that is so vaguely defined?
Remember, it's not. You're looking for SARS-CoV-2 infection rate, not the disease, right? That's the big thing. The vaccine is for SARS-CoV-2, not for COVID-19.
But they define the case as a positive COVID test, any of them.
No, it's a SARS-CoV. No, we're talking about the detection of SARS-CoV-2. What we're talking right now is SARS-CoV-2. Yeah, maybe you're
talking about it now, but
so COVID-19 and the lung and the inflammation and all that kind of stuff, that's second order, right? That comes from a high viral load when your body doesn't have the antibodies in order to deal with it.
Okay, I have one last question.
Sure.
About the when they actually injected the vaccine. I mean, I understand that the idea was that the stuff you inject, it should stay localized, right? You don't want it to spread throughout the body and produce spike proteins like in uncontrolled ways, like too many basically for too long. So it's assumed that it would basically stay more or less in one place, like in the shoulder, and that's why it had to go into the muscle, right? So you don't put it in the vein. So it's a
Yeah,
intramuscular
it's
kind of thing, right?
yeah, basically there's a known amount of RNA that creates an immune response, and that immune response creates the antibodies, well, the storage of the antibodies in your immune system, and then those immune system creates those antibodies in the future. Yeah.
Yeah. So it's yeah. So if it's so, I mean, if it accidentally goes into the bloodstream, there would
Mhm.
be a problem, right?
I mean, no, I mean, there's like the blood-brain barrier, so it's just,
Yeah,
it's
but it
not
would go to the backbone and other organs. Like they have found it like in all sorts of parts of
Mhm.
the body. That's not a
Yeah.
secret, right? It
Yeah,
does
yeah.
go there sometimes,
Mhm.
and I'm not sure about the blood-brain barrier, maybe, but if it goes, I mean, I understand if it goes to too many places, like it's not designed to be at all those places and produce the spike protein everywhere.
Mhm.
So it might overproduce and create a disease rather than immunize you. Yeah? So if it's so
No,
important,
because it's not. So one, it's like a known amount of RNA, and then two, it doesn't replicate so it's not going to be able to create a higher viral load than the one that is delivered, right? Because there is no full like coronavirus there that can replicate inside your cell.
okay, so basically the amount of spike protein produced is proportional to the stuff that's injected.
Exactly. Yeah, it's like
Okay.
25 micrograms of RNA, and we know that and there was
Okay.
actually in the trials, they actually tested different amounts. That was like 25, 10, 5, and then they looked, and
Mm.
I mean you can see all their data. They published it. It's actually really interesting.
Okay, but then I guess like maybe you're basically telling the body to produce the spike protein for a certain time, right?
Correct.
And that time, I don't know what it depends on how this is determined and
Well,
maximized.
we know, right? Because the template of the spike protein that's being made is the RNA, and once that RNA goes away, then there's no way that spike protein is gonna be made, and that RNA gets degraded by RNAs. So are ways to make are in your body just as like a cleanup mechanism.
Yeah,
So
yeah.
RNA is inherently unstable, and then on top of that, the RNAs are everywhere because your body doesn't want like stranded RNAs going around because it's going to produce proteins that it doesn't want. So it's actually very unstable. And actually, this was the problem that actually took from 1990 to about 2015 in order to figure out how to actually protect those RNA molecules long enough to actually create enough protein, because it's just gone almost instantly. And it was actually it's the there's the three
The
prime
lipids.
tail. It's like the three prime tails. It's really interesting. There's a promoter region, and then there's the gene region, which is the spike protein in
Mm,
this case, and then there's the other one at the end. So it's not translated. It's called the UTR, untranslated region. And
mm,
so the idea is like, well, why even have that at all, right? You have the promoter, which is where the machinery binds, and then you have the protein, which is like the real money part of it. But then there's like this long tail part that's not translated. Like, why even have that at all? And well, we know that basically RNA can actually fold 3D structures. So it
mm,
actually kind of like has a 3D protection mechanism in order to protect. They're called like exonucleases or endonucleases to come and like that chomp up the RNA that it has. So
mm.
it's used to protect it. And this is
Um,
one of the innovations that happened, and that's why
yeah,
it was such a long
yeah.
process to
No,
actually develop these things.
interesting. Yeah. Did you also read that paper? There was at least one paper that claimed there was, I'm not sure what's it called, like some reverse transcriptase thing, I think, or something like you basically that the cells
Hmm.
um, learned to produce the spike protein
Yeah.
continuously.
Yeah, so reverse
Like,
transcriptase
uh,
is a way that
yeah,
like is made basically from RNA to DNA. So normally the molecular motif for all of biology is DNA to RNA to proteins,
yeah.
and reverse transcriptase is something that's kind of like in your cells at really small levels, and viruses, retrograde viruses, basically, in order to like put DNA RNAs into DNA. So it's like
So
super, super low amounts. And what happens is
okay,
that those cells basically don't produce it going forward. They basically don't have, because those pieces of DNA are going to be going in randomly into the genome, and it's going to disrupt things. It's going to like mess up cellular processes, so that they'll actually be kind of like they'll be spread out. And that same paper never showed any heredity. It's just the idea that yes, like DNA, if you put DNA into a nucleus, there is going to be some type of, it's called non-homologous end joining. So when you have a double-stranded break in DNA, there's two pathways: homologous recombination, which is basically your body, the nucleus checking, okay, is this control F on this? Is there anything here? Control F on this. Is there anything here? Okay, and then it looks at other pieces of chromosomes and lines it up and
mhm,
is like, okay, this needs to be here. This needs to be here. Okay, we're good. Another route is called non-homologous end joining. So non-homologous end joining is when you just have this piece of DNA, this piece of DNA, and boom, put it together. That can be inherently bad, right? Because you have
something new.
that can be like an insertion, that could be a deletion, that could
Mm,
be, a frame shift. There could be tons of problems that happen, and basically like the cellular processes will kind of like eventually come through and like double check the homologous recombination and
okay.
double check the template and kind of like fix it, and
Yeah.
it'll just work its way out. It doesn't actually like stay,
So
and like heredity
hopefully
doesn't
it's
work.
small the amounts. I
Yeah,
understand
it
that.
happens
That
all the time in your body, man. But your body has a curing
okay.
mechanism in order to
Yeah.
do that.
But the question I really wanted to ask, like, it was related to this injection.
Uh-huh.
Like, why did they make it illegal to check if you hit an artery? Like in I understand in the UK there was
Uh-huh.
an explicit instruction to the nurses that they are not allowed to do the, I forgot the word for it. You pull up the syringe and see if there's blood, and that's sort of a standard procedure with a lot of other injections.
Yeah. From what I hear,
You
it
know
just
what I'm
wasn't
talking about?
necessary, and it was fine. Like even if it did, then it would been you would still create an immune response, and there wasn't any reason in order to do that. That's from
Okay,
what
so
I
you're
heard,
saying,
and from like
yeah.
my nurse family members when I had talked to them at the same time. But it was just like they're just putting it in, and
Okay, so you're saying it's not a huge deal, like if you get in the blood
yeah, I
instead
mean,
of
your
the
shoulder
muscle.
isn't going to have an artery in the center of it. I mean, so you kind of know it, and you usually like you could get some adipose tissue, or you could get some muscle, or so you do that drawing thing if you're actually inserting an IV into a blood vessel. But this is an intermuscular shot, so why would you? It doesn't make any sense to
Uh,
do that.
what is the word for that? Now, I mean, there is a procedure in a way you basically pull the toy back to just
Mhm.
see if you have accidentally hit a vein, or like
Yeah.
some smaller
But
blood
it's an
vessel. It
IM
doesn't
shot in
have
your
to be an
shoulder. You
artery.
don't really need to do that. You only do that for like an IV, so to see like and make sure you're actually
Okay,
in an artery
all right. So,
or
but I
blood
mean,
vessel.
I mean, there must be some blood vessels in the shoulder too, right? It's not like
No,
there's
but not like
nothing.
a huge one, right? Not a vein, or you
Yeah,
know what I
but even
mean?
if it's a small one, wouldn't I mean they could? I don't know. Like maybe it's not that much of a deal. I understood it was like quite important to get it not in the blood, and that's why I was confused. Or
It's
why this?
because that's where most of the translation happens from RNA to protein, so it's definitely more effective. But I think at that point, like you're just wanting to get everyone with shots in arms, and the efficacy of like not double poking some people or making some of it is probably wasted, and they didn't want to
Okay,
like
well,
worry about that.
okay, but then why? Do you know why they wanted to vaccinate like children or like younger
Hmm.
people under 50?
Yeah, there's a bunch of research to show, and we actually just there was someone on 2 nights ago that actually had a kid with a genetic disorder that was triggered by COVID. So I mean, there's a lot of ways that kids can be impacted, and it's like a way for them to spread it to their family members and to create these daisy chains of infection. So it's
Yeah,
completely
but the number
necessary for kids to get
of
vaccinated.
cases was so low. I mean, like
No,
even the officially published number. I mean, there was
but
like
we
even for
we just
half a
talked
year there
about
was not
this.
one person under 50 in Australia
No,
that
that
was
died or
diagnosed.
whatever. But
I
yeah,
mean,
but
not a
we just talked
diagnost,
about
not
this.
with
You can have a
a case.
SARS-CoV-2 infection and not develop COVID-19. So even if they didn't get COVID-19 and died, they could still be SARS-CoV-2 positive and then asymptomatically spread. Right? So that's the idea, is that even if they're not developing COVID-19 symptoms, they're still spreading SARS-CoV-2. And that's not good, and we have to stop that.
Yeah, I don't know. I like this idea of asymptomatic spread. I mean, that doesn't like. Yeah. Is there really evidence for that?
Wait, is there really evidence that SARS-CoV-2 can asymptomatically spread?
Yeah.
Oh yeah, tons of it.
Because
Are you kidding me? That's the whole idea. That's what we were talking about with the wastewater. That's what we're talking about with the molecular epidemiology. The rate of mutation is so more than what a RNA virus can mutate at. Like, where else would those mutations be coming from, man?
Yeah, no, I mean, I understand it does. I mean, theoretically, it can spread, but I mean, isn't that like just a natural form of immunization? Like if most
Yeah, but
people have
but it's
no
a
symptoms,
pandemic,
then
bro. It's a pandemic and people are dying. So it's like,
yeah, but
that's the
pandemic doesn't
idea.
mean it has been redefined. It's not a deadly disease.
It
You know,
definitely was.
it
COVID-19 was a deadly disease, and 1.2
no, I
million
mean the
Americans
term
died from it. So
pandemic.
that's the problem, man, is that, like, I mean, America was one of the worst in the world. We're down with like Lithuania. Like, it was insane. So yeah, we definitely should have done more. The COVID
Well,
vaccine
no,
is
I was just
incredibly
saying,
safe.
like the term pandemic, because you said you were implying that a pandemic is necessarily a bad thing, but
A pandemic is a bad thing, yeah.
I mean, every flu is a pandemic, right? Like if you have, flu, everyone
No,
gets
I
a
mean,
flu every year, and that's a pandemic.
you have local like outbreaks of it, but you don't have necessarily a global viral recombinations happening that in all those spreads. It's no, I don't, not everything is a
But isn't
pandemic.
that like how we actually get the vaccination? Because we know on the other side of the planet they have this, and we
Hmm.
predict then the strands for the next season based on what in other parts of the world happening. I mean, that's sort of
Hmm.
the definition of a pandemic in
Yeah.
the new one. The old one was much more rigid. If you look up the old definition, like I think 2014 or something, where you had to have a high impact disease basically to qualify as a pandemic.
Okay.
But this was downgraded later on.
I mean, HIV
So
is
I'm,
still
I'm
a
just saying,
pandemic. I mean,
like, I'm not saying COVID wasn't a bad thing. I'm just saying the term pandemic is not necessarily what I've seen
what's a pandemic then?
anymore. Well, the way it's defined now, it's just a global spread of something, really, anything. I
Okay.
mean, it was even, COVID was actually taken off the list of dangerous diseases in the UK
I
fairly
mean,
early
this
in the
the
first month.
WHO and all that kind of stuff. Yeah, I mean, but it's still technically a pandemic.
It is technically because
Yeah.
it's of the definition now. But if
Yeah.
with the old definition, it wouldn't have been. I mean, you would
I
have.
don't think so.
I mean,
I think
well, I
you're getting
guess.
epidemic and pandemic, like as Ashley's
Mm,
talking about in chat.
no, I mean, of course, if you're claiming lots of deaths, then. But that's
We're
the other
claiming,
thing.
dude. I
Like,
mean, come on, man. They died. We know they died.
well, why did the all-cause deaths not change? If
They
you
did. We
lose,
have an expected amount of people that died, and 1.2 million more people died that year.
these are like a few percent fluctuations that happen all the time.
No,
I have
absolutely
looked.
not. That's
If you
completely
look at the
incorrect. How many
Euromomo
people
data,
die from the flu every year?
look at the Euromomo data. This is like a
Look at the United
official.
States. How many people died of flu every year?
I don't know the numbers there.
10,000. So it's not like
Yeah,
2018, like 1.1 million people died of the flu, and then 2020, no people died from the flu, and all of them were COVID-19 deaths. Like, we have like an amount of people that died, and they died way more than the previous years. Like, we have, it's called the expected amount of people that are expected to die that year.
Okay, maybe I haven't looked at the US statistics. I just know Germany, Austria, Australia, UK. Also, like some of the
Yeah,
Scandinavian
UK did
countries, and
awful
there
too.
wasn't.
UK is just as bad as the United States because there's rampant misinformation about it and rampant anti-vaccine.
Well, now just looking at the all-cause death statistics, there's a
Hmm.
European database that
Hmm.
really doesn't show. I mean, it used to show actually. When I looked at it first in the first months, it had a history. It went back 20 years or so, and you could look at the trend, and the trend line was going down. So overall, like all-cause deaths were going down a bit, like 10% every 7 years or so. Like there was a nice slope
Hmm.
that went down,
Do you think
and
that,
now
I
it's.
mean, during a pandemic and people that are masking and staying in social distancing, do you think that the flu deaths would go up or down?
I don't know, if it's flu or whatever. I'm just looking at
I'm just
all
asking,
cause
just
deaths.
answer my question. If people are wearing masks and people are social distancing, do you think that flu deaths would go up or down?
I mean, they would probably also decrease, but you have
Okay.
probably some other deaths
You're right.
increasing.
And that hypothesis is true, actually. So normally there's about four flus strains that go throughout in a season.
Although it's
And we
more
actually
complicated.
and the masks and the social distancing were so effective that one of the strains of the flu actually went extinct. So we actually only have three the strains of the flu now. And it was actually in the flu vaccine, there was actually a discussion in microbiology if we should actually continue with that fourth extinct strain in the vaccine. And people were arguing to take it out. Like, why it went extinct? Why would we need it? And it got left in because we just basically have all these people that are vaccine hesitant now. We don't really trust anyone to be make any sense of vaccines, so we kept it in. And so I'm glad about that.
Yeah. I mean, there's also, they only discovered about 15 years ago the actual cause for flu, like why the flu spreads in winter,
Okay.
not in summer, right?
Yeah, dude. I mean, we're learning a lot about
And
microbiology with like it's basically the next generation sequencing techniques that we were talking about before, like basically are reimagining and allowing us to really understand microbiology really for the first time. So that doesn't surprise me at all.
yeah, but why? Like knowing that it had to do with the humidity in the air. Why was in Australia and Brisbane? Why were they panicking and isolating people and shutting them out and forcing them to do all sorts of things and closing down businesses, even though there's very high humidity, like in
Because it
summer.
doesn't because when you yeah because you're socially isolating people, you're isolating them so that they don't
Well,
spread
they,
for
they, they
your
wanted
community
them to stay
spread.
at home in their
Yeah,
air-conditioned homes with
yeah,
the kids
yeah.
instead of being outside where you don't transmit the
Because
virus,
it doesn't because you can no you can't transmit the virus obviously outside, dude. You
but
obviously
much
it can.
less than in a
No, it
dry
just
climate
dilutes it out,
indoor.
and it's not like, oh, I'm outside and now it's like, oh, we're all good, dude. It's like, sure, there's like UV radiation that exists, and there's the wind and all that kind of stuff, but it's not like some magical cure just for being outside.
Well, Greg, you know that wave that if you look at the all-cause deaths, which is basically like correlated with flu only. That's how they test the flu vaccines, right? You look at the deaths, and there you have a peak around December, basically always, and in summer you have a minimum
Mm-hmm.
of deaths, and that's typically associated with flu, with the seasonal flu, which has to do with summer and winter, and basically the humidity.
Okay, so
So
why
it's
okay,
that.
so why do you trust the epidemiologists that are giving you that data in 2015, 2016, 2017, 2018, 2019? But why don't you trust them with the 2020 data?
I don't know. I might trust them, but I saw contradictions and strange things
The
happening.
same epidemiologists are giving you the same information, bro. Like, I mean, they're not like, what do you think happened?
Well, the thing that happened was, like, first of all, I don't know anyone who died of COVID.
Oh,
I
bro,
don't know.
don't
I
do this.
know
Don't do
people
this. You're just making
who
yourself look
died of.
so bad right now. I understand that you can be skeptical, but like being contrarian and just not even recognizing the 1.2 million
No,
people that died of COVID,
no, I'm not
that's
saying nobody
offensive to
died.
me. That is offensive. Do you know that if you take all the war deaths, all the combat deaths in the United States, All of them combined, of every single war that the United States has ever fought, and you combine them, is 1.3 million people. And we lost 1.2 million people to COVID. So you are completely out of line with that. And it's offensive to me that you write off these families and what they went through. It's disgusting. Don't do this. Don't we?
I didn't say that. I just
You're
said that I
offending
did
me.
not. No, look, maybe you implied that, but I didn't say that. What I said is that I personally don't know a single person directly that
That
died
doesn't
of COVID.
matter. That doesn't matter if
Like,
you die, if you know them or
no, it's
not,
just
dude.
okay. Well, it's just if it's a bit, you know, and also like those a million people. I mean, you're saying it yourself. Like if 10 million die of flu every year, like it's a
No,
10% chance.
10,000. 10,000, bro. What? I said 10,000 die every year from flu,
Sorry,
not
that's what I
10
meant.
million.
Yeah,
I think 10
yeah.
million people are dying from the flu every year, bro.
Um,
Come on,
I don't
man.
know.
What's
I
going
mean,
on? What
you
is
have to
happening
check.
here?
Well, the COVID deaths were about 10% of the flu deaths, roughly, in most countries. When I
Oh my
believe I
God, dude.
remember.
Yes, yes. Yeah. In other countries,
Is this
yeah, the
not
United
correct?
States did extremely poorly. Yeah.
Um, yeah. Okay. I don't know. Maybe I don't know what.
They
How
did.
how,
Look
how
at the
did
per
that
capita
happen?
deaths in the United States. Don't believe me, dude. Just look it up.
Okay, but I mean, to be honest, like what's happening in the US is very strange. For decades, they have done a
I know
lot
we
of
were
weird
extremely
things.
bad. We had a president that didn't think that the COVID was real. He thought the pandemic was fake. He didn't
Now I'm
think
talking
for
the
the
last
first 8 months. He
50 years.
didn't believe that it existed. And then when they did quote unquote lock down, it was just a shelter in place. There was no national masks that were mailed to anyone. When the vaccines did go out, it was very soft. Nobody got it, and we had about 30% of the population that dug their heels in and refused to get a vaccine because they were more uh, ideologically driven than compassionate driven and cared about the other people, and they would rather help murder people than they would actually just get a vaccine. It's disgusting.
But why did that the same thing happened in many other countries? Is for example, in Sweden, like there was hardly any limitations, any no lockdowns, and and it
The per
didn't
capita
do
deaths
worse.
wound up catching up to him, dude. It was a very small snapshot in time, but all the different variations and all the different the variants caught up to them. It wasn't like oh man,
Well,
they
it
just.
didn't really.
Yes they did. Yes they did.
If you look at, I don't know what you're looking like. I mean, we have the database for
I
all
mean, the United
cause
States
death.
literally let it rip, dude. There was like what lockdown did we have? You had probably like less restrictions in the United States than we did any of those European countries, and we lost 1.2 million people from it. So it's like your argument is literally what happened in the United States, and we did one of the worst in the world, even though we were way more
No, I
technologically
give you
advanced than a
that.
lot of these
Clearly,
countries.
like, yeah, you seem to gotten like high numbers there,
Yeah,
but there are many other
because there's a
countries.
lot
Like
of
half the countries.
anti-intellectualism, and there's a lot of people that question science, and that have been people that are pushing disinformation here.
No, I'm just like if you look at the all cause deaths, about
Why are
half the
you saying
countries
all cause
globally.
deaths? Expected deaths. It's expected deaths. I've said it multiple times. The expected amount of people that die every year is a certain amount, and it's way
Yeah.
more. It's not all cause deaths, okay? It's the expected deaths.
Well, it's the number that is published. It's called all cause mortality, and I think it's more intuitive because they. I mean, of course, they show also the expected deaths you can deduce from it, but I think it's a very clear number. So you can't argue about misdiagnosis and all these conspiracy theories. I want to get rid of these kind of things and just look at really the number of people died if that number is affected or not. If it's not substantially affected, I mean that's why it's monitored for decades. It's an important number, and there are peaks every now and then. That's the reason why it's monitored, so people know which areas are peaking, and sometimes that happens. And the COVID years did not seem to peak particularly in most countries. In
Yes, they
fact, there are
did. I'm
like
looking
plenty
at
of
all
countries
cause
where they
death
were
graphs
negative.
right now, and all the unvaccinated people are. It's like a huge jump. Like,
I
what are you
didn't.
talking about? What like anti-vaccine like stuff are you going on, bro? Like I'm going on.
No,
I
I'm not
looked it up just
anti-vaccine.
now.
I have had most vaccines.
It's like some RFK website or something.
No, I'm just
Okay, dude.
trying to
I
be
mean,
objective.
yeah, dude. I mean, I'm sorry, but It's
Okay, but anyway,
it's
like, thanks.
yeah. I'm
It was helpful.
I'm a little. You can't write off the people that died, bro. That is just not cool. It's just not cool to me. It's not
Yeah,
okay.
but it's important to be transparent and genuine.
It
What
is,
did
and
I?
people, and the data is there, bro. The data is there, and it exists, and like, it's just not. I don't.
Imagine, Greg, what if it was a little weird and they died of a different thing? Like they
Okay,
died
okay.
of,
How would you test that?
like, yeah, that's what I'm saying. Like that's
How
where
would
the
you test
all-cause
that?
mortality comes
Okay,
in.
then what would
If
you
you,
test that with?
well, you look at the numbers. If all-cause mortality didn't change, the causes of deaths
Okay, so then what
were
was it?
rearranged.
Then what was it?
Well, there was not basically there was nothing too unusual happening. Maybe there was a virus that was affecting
Maybe there
people,
was a
but
virus, dude.
look,
Like, what's your
in most
hypothesis? How are we
countries,
going to test this?
in Germany, they ran out of patients. Literally, the hospitals have to pay back the government because
Okay,
it had to be bailed
how did
out
those people
because they
die?
had no
How did those
patients.
people die? What's your hypothesis?
Well, my hypothesis is that there was not a significant change in deaths. That
Oh
it
my
was just
God, dude.
relabeled.
So so you had a certain amount of Americans
Potentially,
that
I'm not
die every
sure.
year. 1.2 million extra Americans died that year during the pandemic. How did those people die?
Well, 1.2 died extra on top of how many?
Exactly. Expected. Yeah. So then
On
why
top
did
of
they
how
die?
many? On top of how many die normally?
Exactly.
Yeah, but how many? I think it's
I don't
about
know. It's
10
1.2 million more.
in a population of. Let me just think. You have
It's a
300 million.
percentage rounding error. You think 1.2 million people is a rounding error?
Actually, it is exactly around
Oh
10%.
my God, bro. That is
Look
so
up. Look up how
offensive. You
many
think that
people
it's
die
okay that
in
people die
America.
just as a rounding error and that you're not going to care? You're not even suggesting a hypothesis for how this
Yeah, look
happened?
up the numbers. Like really, look up how many people die in any given population. It's around. I think it's. Hold on. Was it
USA
10 or?
expected deaths by year.
Yeah, look up how many people die in a year in America.
Okay. Yeah, numbers of death, leading cause of death per year right now,
No,
49,000. 49,000 that die from COVID every year. Diabetes,
no. All cause, all together.
number of deaths, 3 million.
Yeah.
So 50% more is a rounding error to you?
3 million. Yeah, that's about right, actually. So if you have 1 million, I mean
Bro,
that looks
are you
significant.
kidding me? Are you kidding? You're worried about the
I don't know
qPCR.
the
Wait,
American
you're whoa,
numbers.
whoa, 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 whoa. You're worried about the qPCR statistics and the
Okay, look up the
specificity of
global
being able
number.
to get 1 in a million amplicons and whether or not we can get 99.999 specificity, and you're worried about the statistics there, but a 50% increase in expected deaths, and you're saying that it's a rounding error, and who knows if
No,
how
it's
could
not a
we ever
rounding
know?
error. I'm sorry. I don't want to offend you. Like, can you just please look up the
I'm
global
looking at the
death?
data that you're saying, dude.
Okay, I didn't know the American data, so we know now. But
Okay.
you probably can see the global statistics, like how
No,
many
let's talk about
people.
the United States. 3 million people died
Okay.
normally. 1.2 million more people died from over COVID-19.
Yeah, I don't know if that's the case. That looks significant. So that something must have
Thank you.
happened then. Yeah.
Thank you. Yes. Thank you. Thank you.
But why is that only in America then?
Exactly, bro.
Well, there are plenty of countries
I'll
who
tell
didn't
you
lock
why. I will
down.
tell you why, bro. Because of rampant misinformation campaigns put out by ideologues that care more about political wins than
What about
science
the
and
African countries?
institutional knowledge, and people that are pushing anti-vax disinformation are killing people. That's why.
I'm not. Like what
How
about
else are
the
they
African
dying, bro?
countries?
How else are they dying?
There were no people dying in Africa. I mean, there were people dying in Africa, but not of COVID. Like they completely refused vaccinations in several countries.
Bro,
They
they don't have
burned
the
them.
population density, dude. Come on, man.
Why would that matter?
in
I'm just saying,
in
like,
an
it's not
airborne in
unusual.
an air transmitted through the air like that. You don't think population density is a big problem?
Yeah, I guess that's a
Yeah,
point.
dude. Come on.
But I mean, okay. I mean, then you would see, yeah,
Thank you, dude. Thank you for being honest. I can tell that you're actually thinking about this and you're engaged in the conversation. I really appreciate you. I genuinely do.
yeah, and I think it's important to be open-minded here.
Okay, thank you. I genuinely appreciate you. I'm not frustrated at you. I'm frustrated as a scientist that like it's just I've seen this, like lived this, and it's like it's so I feel like I'm talking not to you like a brick wall. I feel like we're actually having a conversation here, but it's like over the last 5 years, you just see all these bad things happen, and it's like I'm like screaming at the rooftops here, and it's like just people don't care that other people are dying at these crazy rates, man. It's unbelievable. We have like statues to combat deaths, and we have Memorial Day thinking about all the soldiers that have died and all that kind of stuff. Meanwhile, they have the same amount of people that died from COVID-19, man, and they've died since the pandemic, and we don't talk about it. With all those families, those mothers, fathers, brothers, sisters, nobody talks about it. It's disgusting to me, dude.
But,
Do you understand my frustration?
yeah, I totally understand, and
Yeah.
I'm actually studying why so many of us are not learning. It's very hard
Yeah, dude.
for anyone to change their opinion.
Mhm.
If you have ever noticed, like, nobody, says, okay, that's a good point. I changed my opinion based on that. And
Yeah.
that's something, like if you discuss like flat earthers or
Mhm.
any sort of conspiracy people on whatever on either side, clearly nobody wants to change their opinion. It's like it's very hard. It takes
Mhm.
years, or in
It
religious
is.
discussions, right?
Yeah.
So that's why, and I think the reason is because, like, there's several reasons. Quite often you are sort of associating your identity with your political position or religious position or scientific position or whatever position you
Yeah.
are. So you think changing the opinion will degrade your value in your
Mhm.
society or in your community or whatever. That's a very common stance. Unfortunately, that's one reason why no matter what you say, you cannot change somebody's opinion on something because it's
100%, dude.
not
I
based on
100% agree with you, dude, because being wrong, being incorrect is the first step in learning. And people like because of
yeah, but it's so hard.
social media, everyone has to be an expert on everything. And it's like, it's just not true, man. Like it's
Yeah,
being incorrect is actually a good sign that you're actually engaged with ideas that you're trying to critically think with. And it's like, that's why I don't like bemoan people or like I learned on this thing all the time. Like people says something and I'll look it up and be like, oh, that's a cool fact. I didn't know that, you know? And so I think that nobody is an expert on everything and like that's totally fine. That's it's
yeah, and that's why, like why I like science. I like the scientific method, the real one, and
mm-hmm.
I'm also unfortunately aware of, like, when it goes wrong. Like, it's sort of my field where I'm working on. So it's not perfect, unfortunately. There are lots of
Mm-hmm.
problems with peer review
Yeah,
and
it does.
sponsorship and biases and ethics in general. Like, there's lots of hijacking happening, and most students, most PhD students I talk to, they don't know about the risks associated with or the biases that you're exposed to, consciously, subconsciously, and it's yeah, it's hard to be. We are not really trained to be critical of our own education,
Yeah. I
and
mean, so I it's one of those things where science is, there's everything like personal, financial, scientific for me to just show this whole thing and expose if this is all a lie. You know what I mean? I would be the most famous
that's
scientist in the
stupid.
world. I would be rich beyond my means because I would be like showing there was actually some mechanism that we didn't understand here in biology. And that would actually be a huge illuminating thing. It'd be the same thing if I could like disprove evolution, right? It would be an
Yeah,
incredible thing and I would do it right now if I could, but it can't happen because there's just all the data is out there and everyone's kind of critiquing each other's work. Like when, like for example, when the SARS-CoV-2 genome came out and I did my own independent analyses with Benchling in my garage, I'm able to kind of check this and make sure that it like makes sense. Like if there was
Yeah.
no primers, or there was no way that wait, this sequence doesn't make any sense. There's nothing in this genome for the primer to bind to. Like, what? I would be like, New York Times, brrr, like there'd be so many different ways to show that. There's actually
Yeah.
and then expand that towards every single molecular biologist in the world, man. There's like millions of us. So it's
Yeah,
not.
but
Go
are
ahead.
you aware of the problems also, though? Did you know the Antoniadis paper, like from 15 years ago or so? Sort of that gave birth to the science of science, like like a new field of science that actually studies its own methodology.
Are you talking about the
Because
reproducibility crisis thing? Is that what you mean?
no, a bit deeper than that. Basically, it was shown that, like, in many fields, most published research is wrong. It sounds quite preposterous,
What is
actually.
it? I think you're talking about the reproducibility thing. What's the thing?
Not just reproducibility. No, look up like Antoniadis.
Anthony
He basically
Audis?
started Antoniadis. That was
Artist.
published some, I think it LA, University of LA, somewhere there. I think it was Google sponsored or something, and they looked the group, or maybe him, I'm not sure. I think it was his PhD thesis. He looked at the reliability of the accuracy of publications, of papers, of most papers in different fields, and it turns out that there's a surprising, very high amount of wrong results, which doesn't seem to make any sense at all first. But this, when you look deeper, it actually makes a lot of sense because of the way the peer review works and publication actually works.
I think what you're talking about is the reproducibility crisis. So this is the idea that
it's partly the reason.
Yeah, so basically the idea is that a lot of the results that are published in these journals are not able to be reproduced, and basically
No. It's they don't want to be reproduced because you can't get papers out that reproduce studies. It's hard to find a journal that accepts something that is not original. It's just not sexy enough, and that's a sad truth in the publication.
Well, yeah, of course, because it has to be a novel contribution to science in order for it to be done.
Yeah, exactly.
But the problem with in the paper, at least that I'm thinking about, the reproducibility crisis paper, I mean, basically, they're looking at the protocols and methods. They're not going through and trying to reproduce papers. And if they can't reproduce it, it's like take one on the not reproduce and then go to the next thing, try to reproduce it. Oh, we can't produce this one, go on to the next one. What they're doing is that they analyzed all the materials and methods, and they saw whether or not the methods were detailed enough in order to reproduce that work. So that's the problem, is this is commonly cited all the time that like, like the medical sciences, only 80% of them can be reproduced. That means like, oh my gosh, what's happening in 20%? And then psychology is like the worst, where it's like, oh my gosh, only 30% of psychology papers can be reproduced. But obviously they're not going in and reproducing them and then seeing whether or not they did or not. It's just a meta-analysis if there is enough detail in that materials and methods. So I mean, I can tell you myself, man, I've published like 10 different papers. I can talk to you about what it's like to do that.
I've published about 30, I think.
Okay, awesome.
I'm also working. It's actually my job to check papers.
Okay.
But I'm just saying,
You're
like, there is a norm.
You're an editor at a journal?
I'm an editor, but I'm a freelance editor.
Oh, okay, cool. Cool. Are you like a type editor?
Well, scientific, mostly physics, engineering papers.
Okay, cool. I don't mean to dox you. I don't want to like go like into your, you don't have to say anything, but yeah, cool, man. That's awesome.
Yeah, no, I like learning. That's why I like doing that. I'm just saying, like, so there's a new field like that emerged out of this one paper that's called meta science, right? So the study of science, and it's a thing now, right? But there's also, like, I don't know if you're aware of this paper Jackson, where they published a problem with the flu vaccination testing method that has been used for decades. So they found that the way the flu vaccinations were tested was possibly flawed with a very simple test, because they just did the same thing in summer when there was no flu, or supposed to be none, and I found sort of the same results. Mm-hmm. So it seems to be some other effect. It doesn't mean that it's not working at all or something, but it's a weird result. And what was shocking, though, for me personally, and for the researchers, I guess, too, was that they were completely shunned off the community. Like they had huge problems publishing the paper. I mean, this was like University of Edinburgh or Leeds or somewhere in England, in the UK. So they couldn't publish it for a long time. Then once they published it, they were like treated like the bad guys in science for they at conferences. They were like instead of being like, I mean, I understand that it maybe makes you feel uncomfortable if you have been testing flu vaccines in a way that may be faulty. But you still want to engage in a like they may be wrong, or they're most likely wrong, right? If it hasn't been reproduced, for example. So, but this is not what happened in the scientific community. It was literally like, you're the bad guys, we don't want to have anything to do with you, and they were just getting in trouble.
Yeah.
So if you want to publish something that goes against mainstream opinion, or like something that has been done for a long time, and maybe was working for a lot of the science sponsors, and then you find something that works sort of against that sort of view, that's a problem. You get a huge amount of publication
I used to think this honestly before the pandemic, and then it was pretty obvious that there's so many journals, man. There's so, I mean, sure, you're talking like Cell, Nature, and Science, maybe, those get weeded out in the peer review process. They're going to be completely arduous. They're going to be super tough to get into, and they're going to ask you to do a bunch of extra experiments in order to prove what you're right. But there is a ton of journals out there, man, and really people don't know. The layperson, I mean, dude, how many people have you ever talked to? And I've talked to a lot of like conspiracy minded people. How many have they said, oh, this is published on PubMed? And I'm like, published on PubMed? Like, what are you talking about? That's not a journal, dude. They don't even understand. They think a DOI number, it means that it's published, and you look at it and it's a preprint server, and it's actually not even gone through peer review. And like, so there's a lot of like people, we're talking like inside baseball, and I understand what you're kind of like talking about, and the idea of like the ivory tower complex of academia, and like,
but don't you think
for sure that exists, right? For sure. But like, to the layperson, dude, that is not true, like, at all. You can like disseminate something, and it could take off like on social media, and it would catch on, and other people would replicate it, and then they would try to replicate it. Like, what was the paper that there was like a you probably know more than me because you're in that physics world. It was like some like gravitational like floating thing that like someone was like, oh, we figured out how to do this, and that was like all the buzz for like 2 weeks, and everyone was talking about it, and then like nobody could replicate it, and it was found out to be a mistake, and then everyone just moved on. Do you remember that?
Yeah. I haven't heard of that specifically. I'm not.
Yeah, it was like this anti-gravity floating thing that was like, everyone was like talking about it, and there was videos of it. They had figured it out, and then like it got everywhere, and then everyone was trying to reproduce it, and it didn't make any sense. And then it was like, oh yeah, actually it was they actually had like a trace element like in there that was actually creating a magnetism, and they found that it would actually, that's why it was. And so like it does exist, man. Like,
but that's actually a good opportunity to show how science should work. You have to have wrong publications. If you don't allow wrong publications, you can't get right ones. Like you miss, let's say you miss a lot of possible breakthroughs, because Einstein couldn't have, I mean, like all the breakthroughs, if they were like peer reviewed away, which is actually, by the way, I'm not a big fan necessarily of peer review. I think it's, it's actually, it's a fairly new thing. It's only about 30, 40 years old. It's an Elsevier invention.
Dude,
And
I actually, man, dude, I love this conversation. I totally agree with you. And I actually am really frustrated with the peer review process personally. Like, it is very frustrating, and honestly, it's because it's a labor problem. You're getting free labor from
yeah,
these scientists,
absolutely.
and they're unpaid, and it's unfair for people to do it. And by the way, it's pushed on professors, but guess who winds up doing it? It's all the PhD students and the postdocs.
I
So
know, I have done it.
it's ridiculous.
Yeah,
So,
yeah.
so I totally,
And then
totally agree with you. I do think that there should be, then there's some journals that are trying to open up the peer review process, kind of like a Reddit style, like pause, up vote, down vote on like questions of about
I
it.
think archive is good.
Yeah, bioarchives is another one, a preprint server, so that people can like see things before it's
Yeah.
published. And that's another thing where people can talk about it and be like, hey, you kind of need this study, you need this experiment in order to prove this. But and it also helps them, by the way, because it kind of like lays down the stake of like, hey,
Yeah,
we're actually
yeah.
working on this and we're like 90% done, so you can't really like do this study. So I think that
Mhm.
that's a good way of letting people know that, hey, don't waste your time on this. We're going to actually get here pretty soon, because I haven't seen something on a preprint server, and I got scooped once and it was awful. It got 9 months of our work got completely scooped, and we were it was awful.
Worse. Yeah, I mean, you
So
can always publish somewhere else, or like if
no,
it's not
we
original.
tried. We had to add a bunch to it, and honestly, dude, it was. I'm not going to name it, and just because I
Where
don't
did
want
we
to
go?
start
For
drama, but it was
reproducibility.
it was one of the CNS papers, and it was the molecular biology that they used in order to validate their claim was awful. And I was actually really frustrated that we got scooped by something that was so badly proven, and we spent the last 3 months trying to validate the claim that we had through multiple
Mhm.
lines of evidence, and it got published with only one that was like the worst one, by the way. And I was so triggered, and because there's so many problems with it. But
Mm.
it happens, man. It's just you're right. It's there's a big problem with it.
Well, a big problem is that people have to pay for it most. If it's not published in an open source journal,
Yeah.
and it costs you like at least $1,000 or something like
Oh,
that.
easy. Dude, it's like for 3 to 5 thousand most of the times.
Yeah,
It's
yeah,
awful.
I know, that's amazing. I mean, how is this, I mean, a thing? If you think of like where the money comes from and why would people
Yeah.
have to pay double, right?
It's
It's just
so
not.
much money. It's
Yeah,
and you have
it's
to, like, I
it's
mean, you can write it into your grants, right? That's kind of like how people do it. But yeah, man, like it's ridiculous the amount of money that people get charged for it. And I don't know. I mean, the real problem is these journals. Like, their journals are taking in
exactly.
so much money, and
Yeah,
everyone wants to have their stuff open access right now, because you just get more
yeah,
publications from it, and you get more
yeah.
citations. I mean, so
I mean,
everyone
why
wants
would
that.
you want?
But
Yeah,
it's
you
like
don't want your
five to
paper
seven
behind
grand.
it.
It's like 5 to 7 grand, and some of the bigger ones, we're talking 8 or 9. So it's
Wow, I didn't know it
insane.
was that high. Last time I
Hmm.
checked, it was 1,000 or 2,000 something for some of them.
No,
But
I looked in
yeah,
it for one of our CNS papers. We looked into it, and it was just insanely expensive,
how do they justify that?
because
Like there's no.
you're getting one that could make your entire
It's
career. It's
marketing.
Yeah,
I know it's marketing. It's just
yeah,
a fictitious marketing value basically, which you can get easily if like the physicists worked around that with the archive server now.
yeah,
Like you really I mean everyone who cares and everyone who wants to can access those papers and not pay anything and
yeah.
like even talk to the scientists directly.
Yeah,
Like
it's a thing where you yeah, Amy, it's just a thing where you can, if you want to if you want your publication to be open access, basically the journal needs to justify that cost, that it's not going to get through a library subscription with someone, so you pay more money for it to be published. And
I mean it's really a
that's
gatekeeping business they are in.
Yeah, they're making
Like it,
so much money off of it, and it's so
yeah.
crazy. And on top of that, what are they? I mean, the real question is, like, what are they actually paying? What are they getting paid to do? I mean, you have the type editor, you have the
They have
actual
no
artist.
expense.
So the artist is gonna actually take your like PowerPoint figures and then make them all nice and like really like do that. And then you have like your figure artist who's gonna change like it around and make it kind of like
I
amenable
didn't know they
to.
do that. Like, I
the
don't think
thing.
they do any of that
No,
actually.
they do. No, yeah, they do. I
I mean,
mean,
there's,
I've
I
had
mean,
it
maybe they
happen.
used to,
No, they
but
have it.
like, I mean, like as an editor, like I'm supposed to make sure that it's already following the journal guidelines like accurately, and most of them don't do completely, so they will have to do some post editing there.
Yeah.
Definitely, fix some words like how you refer to references and figures and stuff.
Yeah.
And
No, I'm talking about like
but
the
so
vectorization of stuff. I mean, I can send you my I actually have my papers like up, and it's a Nature nanotechnology paper. I mean, I can say that. So it's
yeah.
a review that we had in Nature, and it was incredible. It was a really cool opportunity, and like they made my power. I mean, most of our figures are just made in PowerPoint. It's not like a big thing, but they had the
Really, I
artists
didn't
like
know
come.
that's a possibility to submit it like PowerPoint.
Yeah,
Uh,
PowerPoint, because you can vector. You can
oh, I see. So you, I mean, yeah, okay.
since
I
it's
mean,
vector.
it's been a while for me.
Yeah, Made in Mayhem. I have all my stuff. I'll post it in the show notes for you guys. But yeah, it honestly made it look really cool. I loved how it looks, but it kind of like redid some of the figures, which is kind of weird. But
But don't you think this could be automated already? Like you don't really need like people
sure,
for
for
that
sure.
much. I mean, it's. I mean, they certainly have a software. I mean, you can just tell GPT-5 to make the software in a half an hour.
Yeah,
to
redo
actually do
it,
that
and
job.
I mean, if it can,
Yeah.
yeah, for sure.
So I don't think there is really a justification other than this business of gatekeeping and maintaining an artificial value. Like it's really just like an actor has a marketing value basically, or anyone who is this marketing business. So, but I think it's time to get around this, and hopefully more people start using.
I mean, I'm with you. Like in general, I do think that it's. I don't know what the next step is, and like, and it it's circular, right? The grants want to see publications, and the professors that vote democratically to give tenure to other professors want to see publications. So it's all
Yeah,
kind of like this circular thing where they all
it's
want.
messed up actually.
Yeah.
That I have a problem with universities in the sense that they are supposed to be teaching institutions, but they are primarily like every employee, especially the higher level ones. I mean, literally everyone really is assessed based on their publications, like you say. They are not assessed based on their teaching ability, and those people are still supposed to teach the top level, students in the country. I mean, this is like it's so stupid if you think about it. Like no company would hire like people who publish. It's a completely different skill set. Like doing good research and explaining things to students. Like,
Yeah.
I mean, that's why we have so many bad professors. Basically, they're not.
I put all my show
Yeah.
notes in the threads. So threads, and there's my username, and then today is like August 11th show notes that I put my paper pile. So that's like all my things for my publications, and then
Nice.
my Nature nanotechnology reviews in there if you want to check it out. But
I might actually have a look. When I
yeah,
have
dude,
time.
pretty
Yeah.
cool.
Yeah.
Yeah, dude,
Well,
I think it's like
thank you so much.
inside baseball. I love the idea of like improving it. I love the idea of making it better. The problem right now, I feel like, especially in the United States, is that there's just a huge institutional burning down the house approach. And it's like, and I agree that it could be better, but at the same time, like, I don't know if I would publicly be going out there being like the peer review system is broken and we need to fix publications and stuff like that, when there's like these people that fundamentally don't even believe that science should exist as an institution. And that is the main problem that I see. And it's like, I would love to do what we're talking about, but I feel like those times are dead, man. That was like
Yeah, but
previous.
well, we had a chaos for many decades. Like there has been no accountability for like 70 years in the US, especially. Like nobody ever got jailed or like significantly punished for telling lies publicly,
Yeah.
either as a scientist or as a politician or a business person. Like there's no consequence, or especially like the media. In fact, the media
Thank you,
protect it.
Maiden.
Do you know that there are laws that protect
Yeah.
the Smith-Mundt Act, 1948 was the first version. It was updated in 2002, I think. Like that basically legalized lying to the media. If you look up, that's unbelievable. That was just the way they sold it, as a good thing, brilliant. I mean, there's like a lot of skill involved, but in practice, you basically have no reliability, no accountability, and there are like other bills out there that protect people
Oh, interesting. Yeah.
who
I
lie.
figured out the Wikipedia. So the act was developed to regulate broadcasting of programs for foreign audiences produced under the guidance of the State Department, and it prohibited domestic dissemination of materials produced by such programs as one of its provisions. The original version of the act was amended by the Smith-Mundt Modernization Act of 2012 under Barack Obama, which allowed for materials produced by the State Department and the Broadcasting Board of Governors to be
Yeah.
made available within the United States.
That's the first time inside the United States.
Yeah.
Before, it was designed to create a picture, like a coherent picture to the outside world, like officially because of Soviet propaganda, right? So basically, you had a monopoly on what America is viewed like from the outside. So it's like a marketing image.
Yeah.
It's but basically in practice it means you have nobody who can show any other thing than the government wants you to show, right?
Yeah, I mean,
So
I feel like the real problem, and kind of getting to what you were talking about before, with the lack of education focused in the universities, specifically. I mean, especially now, again, this is like inside baseball, and I would defend academia and universities to the death now. But I am just saying, like, there is a problem though. It's specifically with the amount of patents that universities are coming out with. It's kind of like a patent mill, not really focused on education so much. It's about like getting these patents and spinning off them into like startup companies, because then they could get like some residual income based off of that. So you get a lot of these.
Well, you don't actually get any money in America. Like
Yeah, they do.
it's different. Like I filed several patents like through the universities, and the only time I got money was in the UK. Like, because that's you basically you get your yearly income if the patent is sold or like bought by some company, you get your whatever share. But in America, you sign all those rights off. Like that's
no. As the scientist, as the university, you do not sign those off.
Maybe at university.
Oh yeah, I mean the city, like the university owns the patent. You don't own the patent. You're on the patent as the scientist, but you're not getting any of the.
You get something.
No,
at university, no.
The university does, not you. So that's the problem, and hence the problem, right? And that's the kind of the perverse incentive. So, like City of Hope, for example, made like hundreds of millions of dollars on the bacterial production of human insulin. So, and everyone was like, oh, everyone saw that and was like, oh, excellent. So now, that's where a lot of these, the idea comes from. But yeah, man, it's like. Basically these universities have become patent mills, and as opposed to like really focused on undergraduate education, because I mean, most people don't realize that like the University of California, for example, like only 10% of the revenue comes from undergrad tuition. Like it's not like everyone thinks it's like 70-80%, and these undergrads are making these steep demands, and they're like, grad students, how dare you like go on strike? And it's like, bro, you guys don't run this place. Like, grad students are making, way more money than you by bringing in all these grants than tuition costs. And it's bad. I think it's bad. I mean, in my opinion, I think that like, I mean, if I was dictator of the day, you'd have sports not in universities or colleges. They would not be there because you would be focused on learning, and you would have research institutions, and you would have universities. Universities where you would go to learn, research institutions where the patents and all that kind of stuff goes, and they'd be separated out.
I'm really impressed by what's happened in China the last 15 years.
Mm-hmm.
And I think we should, I wish the world would pay a bit more attention to what's going on in other countries, not just China, but anywhere. Like there are other countries also that have done amazing breakthroughs in all sorts of areas, and how to govern people. I think the Chinese found a quite healthy combination of like a free market society and old style communism. So it's like, I had to change my political stance several times to arrive where I am now, and I'm sort of in the middle,
Okay.
like literally. So and I think, I haven't seen any other country at the moment doing as well, like in literally all areas. I believe there's only one aspect in China, I guess, that makes it unique, is that the sort of the idea, the concept of freedom is quite the opposite there.
Yeah.
I think what happened is that that China has been traumatized in the last 100 years, maybe longer, because it didn't have an effective government. It was, exploited, the opium wars, like there were lots of, abuse basically possible because of it was weak, basically. It was dominated by Britain, maybe the US, I don't know, but mostly Britain, I guess. And lots of bad, experiences, and if you know about the opium stuff around 100 years ago. And then later also there was just horrible things. The population was exploited and not doing well.
Yeah.
And only when they started to be more strict in a way, like the official dictatorship started, that's when they actually stuck to their plans. Like I used to be an anarchist, actually, like completely on the other side. I used to be like a very left also, but then became an anarchist, and that's actually a great idea. But then now I see, like, realistically, it's a bit like modern education. I now also believe we would need more discipline with our kids and in our lives, and a stronger government in the way that is China, actually, because they now have they don't have fear of government like Americans do, or Europe, where you have this the other trauma. You have been in bad dictatorships before. Maybe not in America, I don't know, but in general, like, that was how America was created, because people were running away from these dictatorships in Europe.
Sure. Yeah, it's interesting. Like my PhD lab had a lot of Chinese postdocs and visiting grad students. So it was actually wild. Like 10 years ago, I remember reading this article about like people from China being sent over into nanotechnology labs, and then I happened to join one, and then there's a bunch of Chinese people there, and it was like, and I was talking to them, and dude, because I firmly believe that the United States is the best place, because we do have all the best people from around the world coming in order to stay here, and like as scientists and stuff like that, and we have the brightest minds all coming here in order to solve problems, and I want them to stay. But many of them are foreigners. Exactly, I want them to stay, right? I do. I want them to make families here, and because they're awesome, and they're awesome people. And so anyway, I was talking to them, and they were like, no, dude, why would I ever come to the United States? And I was like, what do you mean? Like the South Koreans, they want to stay? Like, they're like, no, dude, in China, like scientists are like top of the food chain, bro. You get your own apartment, you get a free car, you get you get your own lab fully paid for, like in the city too, where everyone wants to live. Like, it's like, you're great. You're like high at the top of the food chain. Why would I want to live in the United States where it's going to be hard for me to like get a job? And I was like, damn, that's a really good point. Like, I can't really rebut that. We don't in the United States really put scientists anywhere close to the top of the echelon, and that's that's one of the big problems.
Yeah, but it's more different than that, because that's actually also happening, like where I'm now in Finland, for example. Okay. Finland also has been like leading in the sort of education, at least until recently, in Europe and globally, I guess. But it's not necessarily doing that well. And in fact, right now, it's not doing well at all. Certainly not compared to China, which is like literally beating everyone right now in all sorts of areas, from AI to electric cars, and generally lifestyle is amazing at the moment. What I've seen, I want to go there and check as soon as I can. I think it's a pity it doesn't get. In fact, America put out what was like $1.5 billion, like a bill for anti-China propaganda. Like it's just like the opposite of what we would like to see, is like to learn from other countries, whether it's good or bad, but definitely not ignore, especially if there's like 1.4 billion people who are doing really well now, certainly compared to the rest of the world, which is like everybody's like sort of complaining about food prices. And when the TikTok people briefly switched to the Chinese Red Book app,
Oh yeah. Yeah.
and suddenly realized that Chinese are really nice,
Mhm.
and in fact the grocery is very cheap, and it gets like delivered for free to your doorstep, and like it's like all these like things that and then the others, like, they learn from each other for the first time directly.
Yeah.
A fantastic event, actually, a
Yeah.
lifetime historical event happened there for a few days.
I agree. I agree, man.
And I think it's a pity we don't have that again. But I think a lot of people realize now it's other countries may not be the way that they are portrayed, and you have to be a bit sort of critical of the media that
For
you're
sure.
listening to. And everyone, literally, scientists. I'm just that's why I'm questioning. Look, I'm not saying like this or that didn't exist or was a lie. I'm just genuinely interested in the arguments, no matter what. I'm literally going to the flat earthers and want to see the model. I'm still trying to find
Yeah.
an explanation why the stars rotate the other way in the southern hemisphere. And there's something you can see with your eyes. It's very simple, and it's really interesting to study how people answer those questions. But it's frightening. It's frightening not just to see how many people believe obviously in stupid things, because it's more frightening because I know that I'm the same on another level.
Yeah.
Like we all on some level ignorant exactly the way that we see other people ignorant.
No, I think you're really hitting something that's so big and important. I wish people understood how skeptical scientists are. Even when I got a positive result on my experiment, I'm thinking, wait, how did I screw this up, and how is this fake? Like they're inherently in the scientific process, people are very, very skeptical, and it's actually the way we like can reproduce a result. It is like mind blowing because it doesn't happen all the time, right? So I think that being skeptical is inherently scientific. What isn't scientific is what you're talking about, and the fact that like being contrarian. So contrarian just being like whatever you say, I don't agree. Yeah, exactly. That's willful ignorance, right? Because people are willfully not ingesting any new knowledge, and they're staying ignorant on purpose. And that's the problem, is that especially in the United States, there's this anti-intellectualism movement where people kind of just say, like, I had a guy that came on here 2 nights ago, and they was saying that he knew as much about molecular biology as me, and I'm like, I've done it like 16 years as my profession. Like I've done, like, how could you possibly know as much? Like not as like a, like, dude, I'm lording it over you, but like he was saying he was as much of an expert as me on it, and it's like, expertise exists, man. Like expertise is a real thing. Like people really do spend their lives trying to understand stuff, and good effort in order to try to, stand on the shoulders of giants. That's kind of like the meme.
Yeah. I mean, there is now somebody out there that is, I would bet actually, that knows more than you do in your field,
for sure.
and anyone else.
Oh, dude, it's incredible. I go to seminars and I am like blown away.
I think you misunderstand.
Go ahead.
It's a machine.
Oh yeah, well, of course. But like, dude, I genuinely go to seminars and I'm like, because I haven't been a straight molecular biologist this entire time. I've done like, micro host development. I've done plant breeding. I've done nanotechnology, so chemical engineering, like all these kind of things. I've been very interdisciplinary in my background in science. And then I go into a seminar of a dude that's just done molecular biology for like 20 years. And I'm just like blown away, like how this guy is so good, and it's incredible. It makes me want to just like drop my PhD off at the trash when I go out. It's like, it's so cool. So
imagine how it was at the times, like even I think 70 years ago, there was still people alive who were sort of great in most fields. Well, maybe not, maybe 100 years, I guess. Like, the last, what is it called? The poly guy, like somebody who knows about every field a lot. That was possible like until 100 years ago, maybe.
Yeah, Renaissance men, right? That's kind of like the idea.
Yeah,
Exactly.
yeah. It lasted a bit longer than that, I guess. But in I mean, like nowadays, like you know how I mean, we all know about lack of knowledge. Like I know, there are many things that I should know, but I don't know. I would have
Hmm.
to look them up, or I would have to relearn that because, like
Yeah.
there's a certain amount you can keep alive, and then you start forgetting, and it's just impossible, even in a very narrow field. Like I'm
Oh yeah.
not just a physicist. I'm a organic solar energy specialist.
Oh, cool.
Like,
Oh, nice,
you know,
man.
yeah, well, it's a bit boring, like in a way that it's such a narrow field that
I mean, I did
that I
algae biofuels for a really long time, so I guess I'm kind of like I'm on your side, but on the biology side, I'm interested in
yeah.
like chloroplasts and stuff, being those are organic
But
solar
like I'm
voltaics.
also interested in those things. I mean,
Yeah.
I branched out, but more or less privately on the side. Like I studied everything, like from economics to um, yeah, biology or
Have you seen those? Have
yeah.
you seen the chlorophyll photovoltaics?
What do you mean exactly?
They're basically like using the
Oh, to
algae,
plant the
like
solar cells?
yeah, exactly. And
You mean the dye-based solar cells?
I mean,
I mean,
I
they
saw it
have
in
been around.
passing. I didn't understand the physics of it, so it kind of like went over my head. But I could have sworn it was either like it wasn't just a photo bioreactor. It was like somehow using something related to the chloroplast and like in creating that is the
Okay, that would be interesting. There's actually a new science that developed about 10 years ago.
hmm
I wrote a book, actually. What was it? In some book I referred to it. Yeah, I wrote a book about quantum information science, and I think it was quantum.
Hey Amy,
So
sorry.
there,
I'm like
there.
sorry guys. I for chat. Sorry, Amy. I hear you. Like I'm like I when I talk to people, I'm actually engaged in the
Sorry, yeah.
conversation, so I can't really read chat and like moderate it at the same time. So you guys got to like hash it out, I guess, in chat. I really tried to give my full concentration to someone. So try not to take people seriously in chat. If they're, dude, they're not requesting, right? They don't want to actually get involved and actually join the discussion. So they're not really taking it seriously. I don't, to be honest with you, like I'd rather it be there, I'd rather that it kind of I don't want people to be kicked out or blocked or anything based off of any type of like groupthink or anything. We want to be welcoming in this community for people that have differing views, and I actually don't want to kick people unless it's like spamming the N word or something, right? Then it's like okay, we need to like to regulate that, but I don't want people muted just based off of like groupthink and stuff. So that's just sorry. Like if that's offensive to you, like I'm sorry, but don't try to regulate them. Try to get them to join. Okay, try to get them to group up. I'm having a great conversation with Simaga, but at the same time, nobody else is like joining the guests. So these guys need to join up or get they're wusses, right? They're wimps.
Yeah, so yeah, I got also carried away. I'm really enjoying our discussion here. Um,
I'm not
I
coddling
yeah,
them
I
at
was
all, dude. Sorry, Golly. I don't coddle them at all. Have you been here? I mean, that's there. I'm not been. We're not. I'm not. Hi, RJ. How's it going? So, yeah. I mean, just kicking them. Like, okay. So the idea, guys, if you just kick them, right, they're still going to have those beliefs. The idea is to actually give them an opportunity to ask a scientist and engage with the conversation for the first time. So if we just castigate them and we exclude them, they're going to have that same belief and nothing's going to change, right? We've seen this in the pandemic. We've seen this before. And this space is a unique opportunity for anyone to come and actually have their scientific questions answered. So, like I apologize if it's offensive to you that people are being anti-vaccine or something in the chat. Just try not to take it too seriously. Try to get them involved to join up. And like I said, I put up the thing at the top. The queue is empty. Come on in, as a like, hey, get on in here. I'm there's nobody there. So, anyways, Samaga, go ahead. I'm sorry, bro.
yeah. Well, I was talking about this. There's a branch called quantum. Actually, quantum biology is a thing.
Oh,
Have you
okay,
heard of
yeah.
that? Like they a lot of sort of quantum sort of sciences have emerged because they Penrose, I think started with Penrose, Roger Penrose, the
Uh-huh.
physicist and mathematician, Nobel Prize winner, who lately, which I think is a little bit not so quite reliable, his theory about
Mhm.
consciousness, like how these microtubulars could produce quantum effects that could affect the
Dude, you know what's
brain and sort
crazy
of.
is that I totally understand this. Like I've seen this. So with my nanotubes, so not obviously at the quantum level, I mean obviously, but like even for
Okay.
nanotechnology, there's like unique physical processes that
I've
exact
used
that
them
that
too. Yeah.
exist that we don't really understand. And that's one of the reasons why I thought it was a cool way to deliver DNA and use it for my PhD. And I was exploring that. I was exploring basically the different aspects and characteristics of a biological organism, algae in this case, that impact the uptake of carbon nanotubes inside of them. And we're still kind of exploring those
Oh,
uh, ex, uptake mechanisms.
oh, interesting. I was using carbon nanotubes to make solar cells.
Oh, no way. That's cool. Yeah, I know
Yeah.
quantum dots are used a lot. So that's like
Yeah.
I'm not surprised by that. I mean, especially when they're long and they're changed. Do you know how like the nanometer size?
Yeah, I mean, like the ones I had were like like the diameter was like in a few nanometers, basically.
Yeah, yeah.
Like they
I tried
can be
one and
very
then
small.
I settled on five nanometer.
Yeah, in that
And
order,
then
and
they
then they
were
can
they were
be
up to like
quite
about
long, though.
they were about oh man 15 nanometers in length. It was hard. I mean, it's an indirect measurement. We were using AFM to figure it out. But
Yeah, I was using AFM too.
yeah.
It's yeah, it's but yeah. What was my point? You were talking about this. You showed interest in photosynthesis based
Yeah, for
solar cells.
your biofuel stuff.
Yeah, I mean that yeah, that's another a field. Like
Yeah, tunneling. I mean, so
what's
basically the idea was that so my professor was the first one to use plants to use nanotechnology to deliver and showed them uptaking into plant cells. And he
Mhm.
did it with an isolated chloroplast. So my whole thesis was based on, well, if these isolated chloroplasts uptook nanotubes, then would a algae with a cell wall knockout would that actually have similar properties of the uptake mechanism? So it's called the LEAP hypothesis, lipid envelope exchange penetration. And yeah, sure enough,
Mm.
it didn't work. It was actually the cell walled knockouts that actually worked best because my hypothesis is that if you think about a wiffle ball, right, you can actually have like a lot of holes in it, but the cell wall knockout was more akin to kind of like a balloon, where one hole in a balloon and it just pops and the cells just lysed instantly with carbon nanotubes. It was just insane, man. The smallest, I mean, we were talking like parts per million
Oh,
of carbon nanotubes,
hold on.
so small.
You're saying the like the liquid sort of that I don't know the terminology, but what makes up the cell, what's most like that sort of replaced mostly by nanotubes, like
No, it would
instantly.
be just it would
Oh,
just we would add we had the algae, and then I would add the carbon nanotubes that had DNA on them, and then when I added it to the cell wall knockout strain, they would all just lyse, and it would be like they would be popping, so it was insane.
okay, yeah, yeah. I mean, this reminds me a bit on the there were actually the way carbon hold on, what is it, like carbon discs or something
Uh-huh.
are actually used in vaccines. Again, to get back to that topic, like I
Uh-huh.
looked at this at some point because people came up with this ideas that there was what is it,
Graphene oxide.
graphene, I think, like
Yeah.
the like very flat discs that sort
Yeah.
of could be in the vaccines and cause problems. And it was actually interesting to see that this was in fact a thing. I mean, that's a very popular sort of a delivery platform for
I mean, it's
vaccines, it
super.
seems.
No, I mean, no, it's super. No, no, no. The nanotechnology delivery mechanism for the vaccines was a lipid.
Yeah, but
envelope.
there was
So
graphene is considered somehow as a replacement or like some other way of doing it.
it's a contamination. It was a contamination batch issue. So I mean, you could have a spectrophotometer
Um,
figure out whether or not there was like graphene in there, and it would
yeah,
be very easy. So it was just like I am aware of that. I know what you're talking about, and it was just a bad batch. It wasn't like a systemic thing that
yeah.
they just
No, I'm not
lipid.
even talking about the COVID vaccine right now.
Yeah,
I just was surprised to see that it's generally considered a promising platform to deliver stuff.
oh yeah,
Like if you Google if you search in on search of the some of the journals, you find I mean there's so much, thousands or at least hundreds of papers on graphene for in the context of vaccines.
dude.
So
Anyone can join. Anyone can join. I mean, I'm just like we're just having. I mean, usually it's a 10-minute timer, but like I mean,
yeah, I'm
nobody's
sorry.
joined for 2 hours,
I
so
feel a
it's
bit
just me
guilty
and
here.
we're having a great conversation. But usually, guys, when when
Yeah, I
this
don't
like
mind
the queue is empty, when it says queue is empty, anyone can come on in. Like anyone, if you want to talk, like just queue up. It's all good.
Yeah, and a friend asked me to check the papers and the science aspect of these conspiracy claims with the vaccines, and that's how I started looking into this. And I mean, it's a bit similar to your research. Actually, I found there was one paper that was actually looking at how these graphene sheets. I don't know, there was a word for those things. Affect cell walls, and in fact, they found that they actually can scratch or open. I think it was cell walls,
Mm-hmm.
and they were also doing a simulation that showed the same thing, but it required some sort of broken corners of some of the graphene parts to actually do that. But they are potentially they can do it, so that has been shown in one paper.
Hey, what's up, Rock? Yeah, I mean, the size difference is just crazy. The algae to carbon nanotube ratio.
Yeah, of course.
If you're if you're 6 foot, the algae cell Chlamydomonas is like 10 microns. I mean, that's a 5 nanometer carbon
Yeah.
nanotube. So it's like a 5 millimeter pencil lead, and for a 6 foot tall human. So if you think about like, it's actually really easy to penetrate that mechanical pencil into your skin, right? So and think about that as in like you have channels open, you have like kind of like your eye. It could actually get like pretty in like
And it's
a lot
two-dimensional.
Mm-hmm.
Like
Yeah,
it's the pressure on is like infinite, almost like on one side, like on the thin side, you have no surface area basically. Like but yeah, I mean that's just a different topic, I guess.
yeah, it penetrates. I mean, yeah, that's one of the things that we were studying is that when it gets close, basically the carbon nanotube, it'll have such high polarity, so it has to be highly charged, and as it goes in, it basically sucks the phospholipids out of the double membrane, and then it coats the carbon nanotube as it goes in. It's pretty cool. I think I'm gonna make a video about like my PhD like research, and then I'll probably
that
go
sounds
through
very
like do like
interesting.
a 1 minute explainer on all my publications eventually. So I think that would be cool. Hey, what's up, Dr. T? How's it going? So yeah.
We are now reaching the level of LLM knowledge where you can ask them literally to do that already. Like almost
cool.
you might already get something to work quite quickly.
Hey, Rock, I see you.
The
Hey man. Well, dude, thank you so much for the conversation. I really appreciate it. I love that. Thank you for like entertaining, and like I really felt like we had a back and forth, and I really appreciate you going through that stuff with me.
Yeah, I appreciate it too. Hey, thanks,
cool,
and maybe some other time I'm
again. Yeah, you're welcome to join anytime, man. Anytime.
Okay.
Awesome.
Thank you.
Have a good one, man. Dude, Rock, what's up, dude? How's the national lab?
Everything is going perfectly well. How are you?
Nice, dude. All right. How did it go today?
It went well. I had a conference. I made new friends,
Nice.
like-minded friends. Well, I made two friends actually, and
Cool.
one of them is from China. She's so smart, and she's also into like computers and stuff. And I made another friend who's majoring in data science, and we're in a team that we're gonna work together. So we walked a little and we had dinner. We're planning
Oh,
to go to downtown not tomorrow, the day after tomorrow. And I didn't drink too much coffee today either.
nice, dude.
And yeah,
Wait, Rock, is
and I
this like
downloaded.
a girl, like someone that you're interested in?
Not that I'm interested in, but like I'm happy that I could like make new friends. You know.
Okay, cool. Nice, dude.
So
I'm just curious. It's all right. I didn't know. I'm not trying to push you in any other way, or I was just curious. Like, man, dude, that's really, it's awesome, dude. I'm really happy for you.
thank
You're getting
you.
comfortable and having fun with it.
Thank you very much. It was a really productive day. I met incredible people from our connection lab, and
Nice, dude.
talked to them and stuff. Thank you actually. So everything you said, I've been following, and it really works out.
Nice,
Uh,
man.
I didn't drink like too much coffee today. I tried to like take naps during the day, which helped me a lot.
Nice.
Although today was a long day, I woke up at 6, went to get some breakfast,
Uh huh.
and at 7 we like had a meeting, and then at 8 we had a conference meeting. We worked until 6 p.m. But I
Wow.
used
Damn, it was a
my
12-hour day. Jeez.
yeah. But for my lunch break, we had to like kind of compute how these new computer they built in Aurora, in
Mm-hmm.
Argonne National Lab called Aurora. We had to like
Okay.
kind of compute and find out how much energy it like consumes and things like that. So 5 tasks, you know. So I took my lunch break time, which was for 45 minutes, to take a nap, and that kind of
Nice.
helped me a lot. And I was trying to like be calm during the day, especially when interacting with people, which kind of really helped me to like not get anxious and stuff. Uh,
Nice, man.
yeah, it was a really good day. It was a really good day.
Dude. All right.
So I still have like one problem left that I have like to kind of take care of it, which is my sleep deprivation.
Uh-huh.
Even I had like 45 minutes nap today, right?
Mm-hmm.
And then after that, I was like energized, but at night I'm supposed to sleep, right?
Yeah.
But unfortunately,
Usually.
I'm like I have to like go over the modules. And last night I didn't sleep at all. I had like 6 tasks, not like mandatory, right? But I had to
Mm-hmm.
like stay awake, get them all done. So I've been like sleep deprived. It's like in 2 days, 3 hours sleep is like
Oof.
nothing, you know.
Yeah, that's
And
too
like
much,
right now,
man.
like I had no coffee during the day, but I still can't sleep.
Dang. Yeah, dude, naps are so much better than just chugging caffeine. Like I
Oh
usually
yeah,
just like try to like 15-minute naps I'll do like pretty much all the time. Especially if you're doing you're doing stuff that's really mentally draining. So you can just kind of, get that and hit that nap, that little 15-minute nap, they'll change your whole day. I love it.
oh, thank you. Yeah, yeah. I
Yeah,
just
man.
wanted to like join and let you know that I'm getting better, doing
Nice,
well,
dude.
and yeah, I'm really excited for our trip to like my friends and I. We plan to go to downtown St. Charles. We've never been to downtown St. Charles, so we gotta
Nice.
go there, and stuff. So yeah, everything is fine. Yeah. So
Nice,
thank you
dude.
so much for everything.
Oh,
I
dude.
really appreciate it.
Dude, no problem, man. You're killing it already, man. You're doing such a good job. Like I'm so like impressed with your science. I would love to yeah, I'd love to talk to you more about like all your research and your poster, and dude, that'd be so cool to go over sometime. Like it sounds like you're busy and
Absolutely.
stuff. So,
I would love to, absolutely. The other day you mentioned about having like more of like not debate, but like more like discussions of like about some topics, and I had
yeah.
like AI in mind because back in Chicago I did a big project, right? On data science, finding AI biases and stuff.
Mhm.
I presented in different universities, University of Chicago, UIC, different university. Even our congressman invited me to like meet him. Met him. He was like very,
Nice.
so I would love to like if you are
Yeah,
like planning to have debates and stuff. I would love to like join and
for
talk
sure.
about like AI and stuff.
Yeah, dude, I think the idea is to show that like academics have like tons of debates because people really think that like science is just kind of like everyone agreeing with each other like it's a hug box, but like I don't want like toxic debates, but obviously, but like trying to show that yeah, people can disagree amicably, constantly in science, and to kind of like showcase that it would be like really cool to have like a couple of experts Just like talking about like a subject, everyone would know what the subject was. You could even like, and then each side would have their open notes so that everyone could kind of understand what everyone's talking about. And then you could think about ahead of time, like research the counter arguments, just trying to, keep it high level, keep it fun, keep it academic, but also
Yeah.
like really informative too.
Yeah, absolutely. Yeah, that sounds great. I'm really interested in.
Cool.
Thank you so much for your time. Just wanted to keep you updated,
Yeah, dude, I
and you
love
have a
it. I
wonderful
love
day.
it, man. I'm so happy for you. Like this is so
Thank you.
cool. Thank you for all.
Thank
You're
you so
always
much.
welcome to join, and yeah, please keep joining, dude. I love the updates. It means a lot to me.
Yeah. Sure thing. Thank you so much. Thank you.
Nice, dude. All right, man. Well, have a good night. Okay. Hell yeah. Nice, dude. Dude, how awesome. That is so cool. That is so cool, man. Oh, that makes me so happy. Good job, Rock. Oh, that is so cool, guys. All right, all right. Debated a flat earther. I mean, I'm not uh, that is one that's so tough for me. I really have no expertise in that topic. But, if I would just ask questions. I would love to. I would love to. But I'm not you know, that's not my forte necessarily, but I would love to talk to one. Oh, what made me get into microbiology? I love to change genes, man. I love to change genomes around. I love to genetically modify things. Um, um, and you can just do that a lot easier with microbes. The problem with like humans is you can't genetically modify them, and mammals are really hard to genetically modify in general. And yeah, it's just really tough. But with microbes, you can do that no problem, and it allows you to really um, learn cool things about processes and pathways and add new proteins, all that kind of stuff. So I love working with microbes. I love working with microbes a lot. All right. Yeah, man. Like, yeah. Molecular biology is so fun. Like, it's really hard. Any other host system is really tough. So like plants, it can take really long time in order to the order of like 6 to 9 months to make a land plant, versus an algae, you can make it in like a month. So and every algae cell is a plant cell. So it's like, why would I work with land plants when I could work with an algae cell? Yeast are just really cool because it's one of those things that are important for protein production and all these different things, and they're just fun to work with. My dissertation is using DNA to deliver, using nanotechnology and single-walled carbon nanotubes directly to deliver DNA to algae chloroplast. Did you like physics in school? That was, man, physics was something that I loved in high school. I tried it in undergrad, hated it, didn't really like physics lab that much. So I just kind of like using biology. I loved life. I love, you know, there's something that's magical, I think. Well, not magical, but you know what I mean. Like there's something just different about life, I think, which is fun. I did not do a postdoc. I went straight back into industry. So I did 5 years at a biotech between my undergrad and my grad work. And then after my grad school, I went straight into a biotech again. Yeah, dude, physics lab in college is really tough. There's a lot of like calculus. It's like very tough. But I don't really get it. But I think it's cool. It just never clicked for me. They're not interested. Dude, flat Earth is a really interesting one because there's actually a huge Professor Dave like did a video on it actually where there's a huge infighting event happening with flat earthers right now where they like a bunch of them went to Antarctica and they said that they found some stuff and everyone they did a bunch of tests and they figured out that there is no flat earth and now everyone is like these were like some of the biggest people in flat earth. They could actually afford to go to Antarctica and do all these tests and stuff and they uh yeah, they don't believe in flat earth anymore. So now they're being called paid shills, all that kind of stuff. Just like in the same way that I get paid, called paid by Pfizer or the pharmaceutical companies for saying, vaccines are safe and effective. So it's all the same. It's all the same, man. It's all the same. It's so boring. Thank you. Thanks, Maiden. Um, it's a totally different world under a microscope. Oh yeah, the microscope is so cool. Really allows you to um, see a whole. It's kind of like snorkeling when you put your snorkel under the water and you see everything under the water. It's pretty crazy. Is that James? Is that the plants plant-based like eating show or movie? I think it's like I mean, in general, I think that people should just eat more protein in general. So I weigh 180, so I eat about 200 grams of protein a day, and I try to get everything else by like plants that I can. I don't eat meat every meal. I don't think that you should eat meat every meal. Um, like treat yourself nice, get a nice steak or something. But in order to get that protein in, then you really need to use ultra processed foods and protein shakes and protein bars and that kind of stuff. And that's what I do. I've never heard I look like Robert Downey Jr., but thank you, I appreciate it. Vasculomics. I've never heard of vasculomics. Let's check it out. Vasculomics. Vasculomics technology platform has been used to successfully in detecting discovery stage tumor angiogenesis. Interesting. Cool. I had no idea. Interesting. What are your thoughts on the Gaza genocide and America's culpability via our taxes? I think that like that region is like really really hard impact by like climate change and people, there's a huge migration problem that's happening. There's um as a scientist, the climate change has a big impact on the nutrient capability of farming there. Even if they were able to with the blockade that's happening with Israel and stuff. Um, North Africa is having a huge problem with their um, their ability to grow food just in general. And there's a huge anti-genetically modified organism campaign in order to stop farmers in that region from growing any type of food that would help. So, for example, like golden rice is a good example where they actually added vitamin A into rice in order to help like with uh, eye development and deficiency in their diet. And it's unfortunate, genetically modified anti GM uh, propaganda like it impacts people in Africa and it creates them not being able to farm use productive genetically modified foods for farming, and it's really sad. What's up, Nico? Nico, what's up, man?
Hey, how's it going?
Good. Good, dude.
Cool. I heard you talking about kind of subjects and what kind of subjects you were inclined for. And
For what? For
so
the
I wanted to be a
academic like
biologist at one
idea?
point.
Okay, go
Sorry.
ahead. Go for it.
I wanted to be a biologist at one point, but kind of got kicked out of a couple departments because
Okay.
my views were too liberal on the subject.
Okay.
And I wanted to bounce one idea off of you and see if we could maybe meet in the middle or something.
Mhm.
But so I think biologists have a very strict definition of life that is arbitrarily strict.
Okay.
And as one example, I consider viruses to be alive, but a
Dude,
lot of
that's so
biologists
funny.
say,
Yeah.
no, dude, you got it mixed up. And so my thesis is that viruses have, you know, genetic variation, descent with modification, right?
Mhm.
And they have a metabolism that is extended to the host. They hijack the host and they kind of run their
Mm.
stuff. And they go through a dormant period, you know, where they're not active. But, you know, they have a phylogenetic tree. They evolve and they kind of have, you know, from like an alien point of view, if you look at the information,
Mhm.
I would argue that it behaves like an organism.
I am gonna be really lame, dude. I totally agree with you. I mean, this is actually one of my pet issues that I agree, like, that academia doesn't hold this, and I kind of, like, succumb and say, "Hey, like, this is important," but this is actually my second video ever on my old YouTube channel, and I debated my friend about this, and we went through all of the academic arguments and stuff that were from the changeover, because it was actually in like the 1970s that they did this. So um, I'm
Yeah.
actually going to show you guys. This is actually kind of crazy. Here, I'll show you guys my old YouTube
And
channel.
I would love to hear it. And I think her name is Ari. I believe biologists do actually argue that viruses are not, or at least there's like an old school of thought
Mhm.
that argues that they're not alive because they don't have this properties of like a self-contained organism with its own
Yeah.
metabolism,
Yeah,
right?
it's because it's when they go into the nucleotide form, basically. They're just kind of like a sequence of nucleotides. So there just isn't a like fully there you know I think the argument can be made that they have a parasitic form, right? Where it's like they're living off of the nutrients and the enzymes of an organism. And I think you could make the argument there. But unfortunately, when they kind of just go into the genome and kind of subside there, like with a retrovirus, for example, like I think it's hard to say that like a sequence of DNA or whatever is going to be a living organism.
Gotcha.
Why
One
does Dave
other,
agree? Most are just thought, well, nature. I mean, that's the whole idea, dude. You have intelligent discontent, man. This is welcome to science, baby. Welcome. Yeah.
nice.
Let me show you guys. This is crazy. So I had a YouTube, my Twitch and my Twitch and YouTube channel is called The Peer Review. And I was going through and basically I would be like a science correspondent for like Twitch streamers. So I would go on, I've never showed this before. Nico, you brought it out on me. But this is let me show you one second.
Do it.
I can't window. Oh yeah, here it is. So do this. Let me add a window capture here. I actually, so when I did this whole TikTok thing like 2 weeks ago, I figured I was just gonna rebrand and like start everything new. And just because it's just yeah, because I honestly, guys, the peer review is an awful name. It's an awful name because it's just I can't even find my own stuff when I try to find it on anything. So it is it's awful for the search engine optimization. Let's see. But yeah, this is my first video was me
Nice.
chatting with an anti-vaxxer. So that was the this is unreal thing. So it was from that like anti-vax Reddit community. And then, can you guys see this? And then, the viruses are not alive thing, which I think is honestly one of my favorite videos that I ever did. And then President Sunday, I did some stuff with. And then this is Haas, who's like MAGA communists, whether or not genes are real. Heem's like a PhD graduate student that I like, and we like talked about science and like medical research because that's what he was doing. And then In Time is like a science or a news show, and they had all these questions about nanotechnology. So I was going through there.
Dude, you're sitting on a gold mine. You got to clip this stuff and put it on TikTok.
So, and then, this is my Inflation Reduction Act. So Biden's climate change bill for like 30 minutes of like going through the, just like through the cost benefit analysis, and then talking to Fabian Liberty about vaccines. And then I talked to Rob Noor. I've talked to Rob Noor about a couple of times actually, on Lab Leak, and he was like really interested in that. And then Lecture Fan too was a really interesting conversation. But yeah, dude, this is where it all started, where so that's
Nice.
yeah, dude. So it's so anyway, yeah, dude, I think it's a really interesting point, honestly, genuinely. I think that I do see them as like parasites that kind of like live on in an environment of another organism, and they evolved in order to do that. So, and I think that your point about evolution is actually the strongest one, like genuinely. I think the fact that they can undergo evolution, that's a really, really strong thing. And it's not just like a chemical evolution, right? So, like Frances Arnold does do this with the chemical engineering. So she will engineer specific wasn't that crazy? Did you see those the old peer review stuff? Um the the fact that like chemical engineers can do evolution, right? Directed evolution like Frances Arnold does. But the point is that the viral genomes evolve. And I think that is the hinge pin where you're like, if you have a genome and you're evolving, you are like storing that genetic material and you are changing that over time. So I I do think that you're living, like, genuinely. My argument, to the people that say, "Oh, well, it just goes dormant inside of their, like, genomes, and it's just this string of nucleotides, so now it's not, living." Like, I think my best argument against that is, like, just it's very analogous to, like, a tree and a seed, right? I mean, would you say that a tree seed like, a seed is still living? I mean, it requires outside nutrients. It can't be sustained on its own. It can't replicate on its own. So I would argue that it's just kind of a it's analogous to like the seed form of the virus. But then there are bacteria bacteriospores, which are similar to like the seed, that endospores are kind of like a bacterial seed culture, but they're not necessarily what viruses are. So anyway, I mean, the point is that viruses were considered living for until very recently, until the 70s. But
Yeah.
that was also when like archaea was discovered. So it's like like an entire kingdom of microbes were allowed and discovered. So
Yeah, the other topic that I bumped heads with is I used to do conservation ecology during my summers for the National Park Service, and so we counted a lot of sea slugs and kind of just recorded the data. But there was at the time, this was in 2010, they discovered a sea slug that had algae embedded in its back, so it kind of had little solar panels that were built into it, right?
uh-huh.
And at the time, I was like, "Oh, cool, a plant animal." And the marine biologist is like, "No, it's actually an animal that has a symbiosis with algae." And I looked
Uh-huh.
into it, and there's horizontal gene transfer. Like the
Yeah.
the slug is actually producing proteins to maintain the algae, the chloroplast, to kind of maintain all of it. And so, but it's not a full endocytosis like how mitochondria got into the cell, but
Uh-huh.
it's done horizontal G transfer, right? So it's done
Yeah.
some level of integration. And so,
Mhm.
but I always felt like biologists were like, no, it's a plant. It's an animal. It's not a plant. But I've
Mm.
always thought this is. I think we should remove these boundaries and have it be a plant animal, right? Or at least something like that. But I always found biologists very uncomfortable. And I know that the roots of biology come from classification, which, when you're classifying the animal kingdom, I know it's very useful to do that, but
Yeah, I mean,
I
not
do,
everyone's a taxonomist, though. I mean, it's like,
right.
everyone. It's kind of like, um, I but yeah, it's it's tough, man. I do feel like, especially during the pandemic, we actually hurt ourselves a little bit by not like viruses not being living, because it's like, yeah, dude, it's like, people would, I feel like, would have been more inclined to like, like, oh, we got a common bad guy that we can all fight. We can all talk about and fight. And it did feel like viruses were living, right, with the different mutants coming out, the different strains coming out. It just felt like there was a real bad guy that we were all coming towards and fighting against. But um, yeah, it's tough, dude. Like, it's tough. This is sorry to do this. I just have this up. This is so funny, dude. This is such a trip to go through. This is the so yeah, dude. I would go through. This is 3 hours. This is my old. Oh my god. Well, I just got to visit. This, see, dude. This is. I just showed this for a second, and now I got the live taken down for visibility. It's already gotten taken out. This is what I'm talking about. Like, I just showed this for a second, and like, so I would go through biobot data. This is like vaccine data. I would go through all these papers to show like whether or not, like, where this is, and we would just go like point by point, paper by paper for hours. And this is like all I would do. This is back during the pandemic 2, 3 years ago when I'd buzz my head. So my wife like hated it, by the way. But I was like just buzzing my head. But yeah, dude, I would just go everything, point by point, paper by paper, talk to them, like, this is what's going on. And then at the end here, I was actually going through, this is the actual trial data for Moderna and the mRNA vaccine, and it's like that's what I used to do, and then, there'd be what, 256 views over 3 years. Like, nobody wants to watch that. But yeah, guys, that's where I was. That's where it all started. Me doing the peer review. It was
Yeah,
good
we were
times.
locked in. There was nothing else to do. I was also
Yeah,
pouring through
exactly,
that
man.
data. Yeah,
Exactly.
that was a really interesting time because I think it was the first time we were actually tracking all that data too, right? Like that's not a
Yeah,
normal
well,
thing
the
to map.
biobot data, like in the wastewater, was really interesting to follow, because it was just really gives you, like, that. God, it's so frustrating to, man, I show something like with a graph or and it's just TikTok immediately like takes down the search. It immediately pops up and it's like, we've restricted your live, and I'm like, like, how? I'm just showing a graph, dude. Like, how is this?
Did it
It
do it
is.
because of the subject matter, or is it any
No, I
graph?
don't think so, man. Like, I mean, how we talk about COVID and vaccines. Although, to be honest with you, I have tried to promote some of my like my anti-vax responses and like actual like scientific information, and it has not. It's gotten denied on promotion before. So it's like I can't even like promote my own stuff sometimes. And it is kind of weird. But my YouTube stuff has never gotten like taken down.
Yeah.
My YouTube stuff has never. And I've
The
been
problem
talking
with
about
YouTube is the YouTube algorithm is archaic, man. It just
it,
doesn't.
dude. I mean, it's doing something, guys. I mean, the algorithm, as much as I detest like the fact that algorithms are running our lives, like I mean, it's doing something. There's so many physicians, MDs, PhDs, ODs, everyone's like, it's crazy. So it's crazy. There's something going on that got you guys all in here really quickly.
Oh, the
Since
TikTok algorithm is awesome. Yeah.
I know it's amazing. So, um, yeah, dude, I don't understand exactly, man. I don't understand. Like, I'm literally showing graphs. Like, how would it be? Like, people say, oh, it's because of the logo. Um, that would be cool. Thanks for sharing it. Yeah. So honestly, if you guys can um, share it, that's the best thing. Cause I
Do
don't get
you
discovered sometimes.
do you buy into the RNA world hypothesis?
Oh yeah, for sure, dude. I think
Yeah.
that's the thing that makes the most sense. I mean, because RNA can create enzymatic activity. That's the thing that people don't understand. It's like, oh, people say, oh, you need proteins in order to make RNA. So how can RNA world like exist? And it's like, oh, well, I mean, RNA can fold and have enzymatic activity. That's the big thing.
But so
Um,
okay, so like
Thank you for sharing, guys.
DNA.
Thank you for sharing it. Yeah, we got TikTok restricted, so it's just us hanging out until. But it's fine. I mean, it's fine. I'd rather we're having fun, and that's part of my journey.
Yeah. Wait, so with RNA world though,
Hmm.
so that is a hypothesis that explains the origin of life, but does it ultimately explain the origin of DNA and like we're patching together more and more strands into one
Yeah,
master strand?
yeah. It's like
That's-
it's the idea that nucleotides could lead to a complexity of life to that like we like see it now. Gary, I see it, man. Don't go away. I'm down to talk to you.
Because it
Um,
seems like viruses are that, right? Like
mhm,
viruses integrate into DNA as well and kind of
mhm. Yeah.
build the code base, right?
Oh, yeah, yeah, exactly. Yeah, retrograde viruses, reverse transcriptase, like it's pretty well known. So, yeah, yeah, that's
Yeah.
cool, man.
Well, right on. I'll let you get the other guests in, but yeah, man, thank you. It's
Uh,
good to catch you live.
yeah, thanks, man. Yeah, I appreciate it. Yeah, Gary, we just do one at a time, man. It's just it's more fun that way. I think it's easier to Gary, come on in, bro. Um, it's I think it's just more fun that way, man, to talk and have like an in-depth conversation, one person to one person.
I'll catch you
Or did you
next
want to
time,
ask
dude.
Nico something?
Oh,
What's up, Gary?
oh, and to actually answer,
Sure.
I think do you Cray asked the algae provide sugar for the slug, so it's kind of like a little solar panel that provides energy. And she asked
Mm.
what the algae do to the slug.
Which slug?
Um,
The algae did which slug? Oh, are you talking about the endosymbiotic algae that
yeah, that's in
oh
the
dude,
sea
that's
slug.
so cool. Yeah, yeah, yeah. It is very cool. I know what you guys are talking about. Those pictures are wow, they are so sick.
Yeah,
They are
it's
so
crazy.
sick. Yeah, dude, biology is so cool, man. There's like stuff that like it just blows my mind. Like
Well,
I think it's
the
so interesting.
one of the things that I worked at a plant-based food company, and
Oh, cool.
we were trying to recreate the symbiosis of bacteria that go on in cheese, but
Mm-hmm.
created for plant-based cheese. But the synergy between all these bacteria is so insane that like you just can't like you know inoculate a plant-based substrate and hope that it all kind of you get a lot of spoilage, right? And
Mm.
so um, I was then looking into other symbiotic systems and realized that a lot of this stuff, I don't if you look at dairy and milk and cheese, like I don't even know how that whole thing just evolved into a food. It was like a naturally evolved food, but it's the most complex food in the world, I feel like. And then even with beer, like beer production is insane. Like how did humans figure out how to control that, right? Or I guess it's a
Yeah.
wild fermentation, so
Yeah,
kind of
I
happens
think that is
naturally.
Gary. I think that is the one. Yeah, Alicia chlorotica. I mean, other organisms like get drunk though. I mean, you have like monkeys and stuff eating old, fruit that's like on the vine that has been rotting, and they'll eat it and they'll get drunk. I mean, a certain percentage of wild microbes, yeast in this case, like make ethanol when they're
Yeah.
in anaerobic fermentation. It's just yeah, it's just you normally have to culture it, you have to isolate it, but yeah.
And it kind of sterilizes the system when it builds up, right? The ethanol.
It usually kills itself off. Yeah, exactly. So
Oh,
that's kind
right.
of the
Okay.
problem, right? Where it's like it'll literally make enough waste so that it just dies from it. And that's the idea with the microbial community, right? Where like one species waste is another one's fuel source, and then they kind of like all create this web of community where they're all giving each other information and everything like that. Gary, I'm really tired, bro. I'm gonna go to bed soon. So if you want to like ask, I'm down, but yeah, like request up, it's all good, dude.
Yeah. I don't know if Gary was wanting. I'll let Gary talk to you, I guess.
Okay, sick,
Yeah,
dude.
but yeah. I'll catch you next time, dude.
All right, thanks, man. Appreciate you.
Yeah. Right on.
All right, guys. Well, guys, it's been a sick week. Really cool. I didn't expect to do this today. Let me see my schedule. I'll probably tell you guys what the next one is. Okay, yeah, it's gonna be Friday. Friday. So like Friday night. So if you guys want to put it there. So Friday night, gonna do the stream. Been awesome. I set up a Discord for the subscribers. I mean, I do. It's new, and I'm still kind of like on the edge on it, to be honest with you. I could be swayed one way or the other, but right now it's for subscribers only, just because I want it to be like an actual. Oh yeah. You can go to the pure.review. The pure.review. The idea, I don't want it to be a dead Discord channel. That's the thing, guys. Like, I don't want people just lurking in there. I want people that are actually going to be in there, like requesting articles, posting stuff that, and want to be in there so that I can actually give them my attention for like getting jobs, working on their CV, getting the things going, figuring out like coaching and that kind of stuff. So I would love that. That's the idea. Like if you want like professional or personal growth opportunities, and to be more involved with the stream, and I do like behind the scenes videos, that's kind of thing. So that's what I'm thinking. Thank you. I appreciate you. That's why I'm doing with subscribers only, but that's the idea. I honestly could have my mind changed, but that's what it is right now. So, um, so Friday. Oh yeah, that's DNA, dude. Ooh, isn't that cool? I freaking love it. I love that picture. And I love just like abstract art. So I love the just the random stuff. I don't know. I don't know what it is about it. Just cool random stuff. Let's see. Yeah, it's been a really cool week, guys. It's been a really cool week, just in general. Um, Last 2 streams ago, Dean Withers followed me, which is wild. I thought it was like a fake account, and then like I saw it, and it wasn't. So I was like, whoa, that's cool. I basically have like he's like one of like 2 people that I know on TikTok, and so it was really cool. And like I feel like this week it was just awesome. Like really felt like we're getting something going and really getting the ball rolling on like this community and everything. And I'm really fortunate to have you guys all here, and I really appreciate you. I did put out that video of those guys crashing out. I don't like the toxic videos, you know. I don't want to I but I thought it was funny. So I kind of like baited it. I knew it was going to go a little bit viral. And so I put it out there. I don't know. I'm a little conflicted. I don't want to put content out like that all the time. I do want to put out like real positive content that like is educating or illuminating, and that's not going to be all the time, you know, but it's just yeah, exactly. I think it was it was specifically pretty toxic, both of them, pretty quickly. They both kind of devolved. The one guy, I don't think that he was to be honest with you guys, I'll be 100%. Like, I was not expecting the anti-evolution arguments to be happening like multiple times a night, and I'm very surprised about that. I've been interested in Christian apologetics, anti-evolution arguments since like early YouTube. I was like, you know, the guy from like Family Matters or whatever, you know, he had like a Christian apologetics anti-evolution like show that he like went through and was like talking about it. And it really, dude, what is this Red Waters Worldwide thing? Yeah, Richard Dawkins era, like the guy, he had like curly hair, and bro, I'm just gonna and I've just been really interested in what people believe, why they believe it. And I don't know. There's something about evolution is so apparent. And so it feels weird. It feels weird to be back in life. It feels like it's like 2003, 2004 internet. I'm debating evolution, but I love that stuff. I've been loving it. Oh, okay. Okay. Yeah, flat earth. Exactly, dude. Flat earth, evolution. I'm like, what is this? And so I love it though. It's cool. And I mean, the amount of genetics and all the information that we have now is just wild about it. But yeah, I thought that it's funny to like it's probably like his first time ever talking to a scientist about it. And I actually have my undergrad degree in evolution. So it is kind of wild to just have someone have an opportunity to actually discuss it with someone. And he just yells at me the entire time. My favorite though, my clip is coming out. How did I find you from my YouTube's on my profile. It's like where all my TikTok archives are. But yeah, dude, it's I put out it's the first two guests 2 nights ago, and if you guys wanted to watch the full clip, but I'm putting it out soon as a clip, and he basically said Neanderthals aren't real and that they're just funny looking humans. So it's like, dang, dude, that's like that you really are committed then. So it's yeah. I know he didn't believe that. He thought that like, oh, we created the Neanderthal DNA, and now it's like circular argument thing where it's like, oh, you created Neanderthals and then you conveniently put it into your DNA. And I was like, I'm trying to think about like, do I really need to explain 16S ribosomal sequencing to this guy? There's no way. There's no way, man. How do I do this? So, but yeah, imagine that. But anyway, guys, it's been wild. It's been really cool. I'm really blown away. And I appreciate you. I really do. So thank you for being this new addition to my life. And Thank you for being. Yeah, I'm going to bed right now. So thank you. This new cool addition to my life. I'm gonna oh guys, actually I do want to ask you genuinely. So like this live, for example, has gone on for like 3.5 hours. So like would like a 1 hour nightly stream like work? Most people like my TikTok analytics are like off the walls for how long it is. They're like, "Why are you doing this? Like, what is wrong with you?" And I'm like, like the the TikTok live analytics is like plus 5 minutes, plus 15 minutes, plus 25. That's where it ends. It's like most people stop streaming at 25 minutes. So I guess, and here I am like doing these 3 hour, 4 hour, 5 hour streams. I guess I mean that's what I used to do at YouTube and Twitch, so that's kind of like what I'm used to. But I guess TikTok, I can't imagine going for 15, 20 minutes. Okay. Dang. Okay. I mean, honestly, guys, if it was like 30 minutes to an hour, I could do that every night, like no problem. I genuinely like that. That's pretty easy for me. Oh, thanks, Manda. I appreciate you saying that. Thank you. Okay. Okay, that's good. Duke's Cray. Duke's Cray. Is that how you pronounce your name, dude? Um, these are pretty normal. Okay. 3 hours is normal. Okay. Ducray. Okay, thank you. I appreciate that. Ducray. Thanks, Ducray. It is easy to lose track. I mean, that's the thing, man. When you're trying to really like explore an idea with someone, it does take long to kind of like unpack. And like that first guy that came in here was like over an hour. I do have the timer now, trying to get people going, just in case. And I do the one-on-one just so that we can have an in-depth conversation. I think that I do like that. And I did put the follows only for the requests. I know people hate that, but at the same time, I would like some friction on like requesting to join, just because I do want someone that's genuinely interested in a good faith argument and not just like coming on. So that's what I'm thinking about. The beginning, you need lots of people to discover you, so you need longer. Yeah, so that's the thing. Exactly, juiced. Like that is the thing, man. Like I will go live for the first like 20 minutes and it's like 5 people. So like, and now there's like 48 in here. So that's the thing, man. I'm like, I don't understand how I could only go live for like 20 minutes. But then I scroll through TikTok lives and it's just people like literally staring at their camera going, give me, give me, give me, give me this, give me this. I'm like, how do I don't think that's really my style. But yeah, I don't know. I don't know. I'll try it. Maybe like we'll try an hour. Okay. We'll try an hour like every night or something. I can do like 9 p.m. PST. The kids go to bed at 8. Then I gotta like feed and walk the dogs, do some dishes, and then I can do it. But I can do like an hour a night, and because I got to get up at 4:30 in the morning for jujitsu. So that's like a oh thanks, Ducray. Okay, cool. Yeah, thanks, Manda. I agree too. This is the type of this is what I would watch, right? I'm making basically the content that I would watch, not what I see on TikTok. So tell me, like you can help. You guys use TikTok more than me. Okay, cool. Thank you for the feedback. Um, Interesting. Oh, cool. Thanks, Maiden. Sweet. Appreciate you, Maiden. Cool. Thank you. Yeah, I appreciate it. I think though, because it's debate and 10-minute timer, you'll get it going. Yeah. I do like I mean obviously I put debate in my name, but I find like just like in good conversations fine too. I don't want that. I look through in different things for my I was actually asking ChatGPT, like, what should my name kind of be? I want to do this, and I want to do science communication. I want to engage people, and it's like if you're engaging people, it would be like Dr. Greg Live or Dr. Greg Talks, or I didn't feel like that was a really good way to draw people in. I feel like debates is more of like okay, we're gonna like go in kind of thing. Oh, you like Dr. Nanotube? Oh, okay, sick. What's funny is I actually have my 30-day thing up, and I was thinking about like just moving my Dr. Greg debates into my name and then putting myself as Dr. Greg, but I don't know, so I think, dude, Dr. Nanotube, I mean, it's literally from my username's from my thesis, and I thought Nanotube is kind of like a cool, nanotechnology thing that it's kind of like unique, so maybe I keep it then. Debates can be productive, and that's kind of like the whole thing too. I want to show people that like academics debate all the time, and it's not toxic, and it's a good thing. I don't know. I Guys, tell me if I'm crazy, but like I'm literally debating ideas in my head all the time. Like trying to think about, like, am I thinking about this like the right way? And I try to like think about it from the other side constantly, and I'm trying to like make sure I'm thinking like, is this correct? Especially, oh my gosh, when I was doing my research, I was like, dude, my thesis, I'm like thinking about my dissertation, how many holes I have, what was I thinking? um okay, thank you, Dr. T, thank you, I appreciate you saying that. Yes, 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 man. I agree. Exactly, Maiden. Yes. Okay. Cool, cool. Good, good. I did. Yeah, I published a literature review in Nature Nanotechnology. It's not an original article, but yeah, we did a review. Smart plant sensors. It's all in my I actually, if you guys want to check out my publications, I put the paper pile in my threads. I Dude, someone in the comments on one of the toxic crash outs was like, "Give me, what's your Google Scholar? What's your ORCID? What is your ORC ID? I'm looking you up. Give me your PubMed profile." And I'm like, I've already actually said all this. I like literally have my LinkedIn. I have my paper pile. Um, what is my H index? I don't know. I like my, and it was like, I was like, I don't know, dude. This doesn't feel very. I was like, this doesn't feel good. So, my H index is 6. All my citations are 927. I'll put my Google Scholar in the bottom. Yeah, exactly, Dr. T. I was like, I normally am very about I'm okay giving that information out, but like you seem to not be in the right spot for us, so I didn't do it. No, no, Nico, it's fine, dude. That's like genuinely, if someone has a PhD, I'm actually really interested in their dissertation. Like I'm actually like it's not like a what are you doing kind of thing. Like I would love to like read what you're doing, Yeah, exactly, Gary. No, yeah, for real, dude. Yeah, that's why so my paper pile has all my PDFs and everything. So if you wanted to read any of it, you can. Yeah, for sure. Yeah, for sure. Exactly, Dr. T. And that's what I was like saying before, and sorry for repeating myself, but like as scientists, we are more exposed than most people because we have to have our last name out there for our publications, and then our that also includes our email, and that includes our address because that's where our university is. So it like opens us up for harassment, right? So I made it. Oh man, do you guys want to go through this? I guess you know what? We're chilling. We're chilling. And this is at the end. So if it doesn't matter, I actually made it into the QAnon conspiracy world. I'll show you guys the thing. Oh my God, this is so wild. So I gave, I never pull guard. I will take you down. I will collar drag you and single leg you right in. So, okay. So I am going into this nanotechnology lab, and the professor kind of thought of the chloroplast as kind of like this way of as a sensor for reactive oxygen species, kind of like a motherboard to see what's going on and how the plant is going on. But I came out of an undergrad thing, biofuels lab, where we made anti-cancer drugs inside of chloroplasts of algae, right? So I'm talking to him just like, spitballing ideas. We're in a brand new field, dude. Like nanotechnology in plants is really undiscovered, and we're exploring it and talking about things. And I was like, yeah, dude, I really see like chloroplasts as like 3D factories, actually. Like we could actually make things in there. Sure enough, obviously, the vaccine comes out with like RNA and all that kind of stuff. And I'm like, dude, you could make these RNA vaccines inside of like chloroplasts of plants, no problem. And there's no RNA interference. So it'd actually probably be a good place to do it. So he put a grant out there for the production of RNA vaccines inside of plant chloroplasts. And you'd basically put the RNA on top of, cling that to, or DNA or RNA to the carbon nanotube. You'd put it into a plant leaf, and then the plant leaf would take up that material. And yeah, lettuce or spinach, it's usually spinach, because it's just easier to do it, and they have a really high concentration of chloroplasts. And then you just like make vaccines inside of plants within like, an hour. You wouldn't have to like transform the plant in order to do that. So this got out to Michael Flynn, okay, and that they are these guys, the Illuminati or whatever, which is like me or in my PI, like they're making vaccines into the lettuce at salads. And so that's what this was. Yes, this was me, guys. Jen, that was me. That was me. If you actually remember this was legitimately me. Here we go. Yes, I know. People, it's actually wild. People know about this. Here. Do. I know, isn't this wild? Okay. So this is it. I don't know how to crop this. Can I crop it? Okay, here we go. Okay, here we go. And then he talks about this on this freaking so yeah, like Stephen Colbert talked about it. It got to Colbert debunk Flynn's vaccine salad dressing theory gets at disinformation station. Can you guys hear this? You guys hear this? No. Okay. How in the world do I do this? Anyway, oh my gosh. I will put this in the show notes, guys. But yeah, dude, he starts talking about this. That we're putting vaccines and then there is this show. This is it. This is the clip. I'll put this in the show notes. Check this out. He's going on these podcasts talking about this, talking about our research, saying that they're putting vaccines in salads. It's like, What? What? So this is Michael Flynn. The dude's a traitor. He's a literal traitor. He sold out the United States to Russia, took money from them, was a disgraced ex-general, was caught completely red-handed, being a spy, basically trying to for the Russian government. Completely pled guilty, was like, yeah, you got me. This is awful. And then Trump was like, what are you talking about? I'll just pardon you, dude. Don't worry about it. Gets a pardon from Trump. Anyway, Michael Flynn is an awful, awful, awful person. Okay, so here it is, guys. Here's my threads. Yeah, I know. I'm for real. I totally agree, dude. So this happened, right? People started going crazy. They were emailing my PI death threats. They were giving, like, it was to the point where people had like security at our university, like around the building. They were doing people were being walked to their cars. It was wild. It was absolutely wild. And then I thought that was all over. Okay, I thought this whole thing was over. That got crazy, dude. Well, guess what? Tennessee banned salad vaccines. They wrote a bill. They started banning salad vaccines. So Tennessee. So it's unbelievable, dude. There's like 10 minutes of House debate where they're talking about how dangerous it would be to have salad vaccines, like vaccines in salads at supermarkets that you don't know, how dangerous it would be. Speaking of the research, she said the idea this would be somehow correlate to some kind of retail offering of vegetables. Yeah, he's like, it would classify any food that contains a vaccine or vaccine material as a drug under Tennessee law. The bill defines vaccine materials as a substance intended to stimulate the production of blah, blah, blah. I was like, what? So then it was so crazy. They started talking about how I'm putting this all in the show notes, guys, so you can look at it. And then there's like Tennessee House video, and I'm watching this, and they're talking about like, we need to outlaw the scientists, these rogue scientists that think they're cowboys. They have no idea what they're doing. I'm like, dude, I might be like an outlaw in Tennessee. Like, they might be coming after me, man. It was absolutely wild. Yeah, here's go. UCR received a $500,000 grant from NSF to study if it was possible to successfully use plant cells to replicate DNA containing mRNA vaccines. Like, it's me, guys. It's me. You would have to get a prescription to make sure that we know how much of the lettuce you have to eat based off of your body type, so we don't under-vaccinate you, which leads to the possibility of the efficacy of the drug being compromised, or we overdose you based on how much lettuce. I love the idea you could overdose on vaccine lettuce. So it's all fear mongering, Gary. That's the thing, dude. These guys, it's like not asking questions, not caring about what the actual truth is. I honestly, I was actually outside of the lab. I'd graduated at this point, and I'm like, do I like go and like email these guys and say like, I can answer questions, like, if you guys really want, like, this is kind of nonsense, like, this, I'm literally from this lab, but I'm not the PI, obviously, so I kind of felt out of my place, and I just wanted to let it roll, like, to kind of die out. I didn't want to like involve my PI anymore. So anyway, yeah, dude, it's crazy. My whole point is that, like, everyone thinks that, Copernicus and Galileo, they're like, oh, that the olden times, or when, the scientists were persecuted and stuff, but I did fizzle out. Like, that was one of the things. I didn't involve myself. If I did kind of like put myself into it, I would have like kind of invited more harassment. So I didn't want to. What's up, bud? So I just kind of let it die out, but yeah. Exactly, Gary. Exactly. Like, as an institution, we're being attacked, like, for real, guys. We really are. It really is no joke. Um, and, uh, and yeah, dude, I mean, this is why, by the way, the same anti-intellectualism, the same institutional distrust that impacts science is the same one that impacts like medical doctors, right? And this is why none of them have any problem with me using like doctor in my name, because it's like, one, I really am a doctor. Two, like, the same thing that they're being impacted, I'm being impacted. It's not like they're, in good faith, these people are being, judicious and trying to really uphold the standards of MDs and physician care and all that kind of stuff. Um, yeah, dude, that's so sad. I'm so sorry about that, Gary. That's awful. And I mean, at the same time, right? These are the same people that are like going to chiropractors for, yes, exactly, Dr. T. It's so frustrating. And I mean, I joke with my MD friends, but it is true. Like, as a PhD, you're literally contributing to your field, right? Novel research. And like, an MD by itself doesn't do that. They're not scientists. They're not like contributing to the wealth of knowledge. Some of them do, right? Some of them do clinical trials, and that's awesome. Some of them do masters and or a PhD. MD PhDs do exist, and it's awesome. I appreciate them, and I think that it's awesome that what they're doing, and we're all trying to give back, and we're all trying to make this world a better place. But yeah, I mean, I joke with my physician friends about them being like mechanics, right? I mean, it's a joke, and we all love, and we're all trying to like, make the world a better place, and it's all good. But but I do understand. Let's be real though. Like, I do understand why the designation exists inside of a hospital, right? You do not want anyone to be confused about who is the doctor in this. Exactly, they're meat mechanics, they're human mechanics, Nico. I mean, it's true. They're physicians. It's okay. That's totally fine. But I understand, like, in a hospital setting, if I was a clinical microbiologist there, right, and I was a doctor, I had my PhD, I wouldn't be sitting there going like, oh, well, I'm a doctor too, but he's the doctor that's making the name, or like the call. But I don't know. I mean, like, what is the correct call then for like a PhD? Then, if because they did contribute to their field, and to Dr. T's point, a PhD is the highest degree awarded in academia. It really is. So I feel like that there is a word that's separate. It's called doctor, and I call like physicians. So I mean, that's kind of like what I've started doing, like especially with my kids and stuff like that, because like, you can have a physician that's not necessarily an MD, and that's just true. That they're actually, you're describing what they are. I'm not and not in a bad way, but like that is what they are. So anyway, I don't mean to disparage people, but I'm getting blown up in my own comments, and that's why I'm talking about this, which is just funny. So anyway, that's just like my thoughts on it. But I love the amount of MDs that are here. There's a huge amount of MDs and physicians that are in the community, and I think that's awesome. I love it. I don't know how so many people, how many physicians found us so quickly. How many PhDs are in here, It's incredible. Like it's crazy seeing all these the everyone's credentials around their names. I'm like, dude, this is so cool. And all the MDs that come in here, what are they saying? Like, oh, I'm finally found a good science stream. I finally found a good place. This is incredible. None of them are going like stolen valor. How dare you get yeah. So it's like, I mean, those poor physicians, man, they have to deal with NDs now, naturopaths, right? That's the craziest thing. Naturopaths are calling them NDs and they're calling themselves doctors now. That is just, oof, that is wild to me. That is now that is dangerous, right? Now it like these people that are so upset about me calling myself doctor, obviously that has to be a step too far, right? That has to be a step that they kind of step in for gatekeeping, right? That obviously is too strong. But it's not, right? It's not. They do not care. They don't care about it at all. So it's this feigned ignorance thing. And these are the same people that are like the death of expertise, right? That don't really, that don't respect expertise at all. And they think that they're as much of an expert about everything as you are, but they're really the ones that are upholding the specificity and the titles in academia. It's so funny. Yeah, exactly. Yeah, yeah. I don't know if she was a naturopath, but yeah, but yeah, she might have, I don't know if she calls herself doctor, but yeah, the water memory person. I really yeah, I don't know. I don't know. That was a really frustrating conversation for me. Probably one of the most for me because it was like popcorning between a lot of like I'm sorry, but if you're bringing like quantum mechanics into like light energy and then you're talking in order to understand like what crystal works best for you for healing, you're like, oh, what in the world? Exactly. Exactly. There's no way, bro. There's no way that like I have a microbiology PhD and I don't even call myself like an expert on immunology or virology. Like and she's coming in with all these different topics going like I know this at an expert level, I know quantum mechanics at an expert level, I know light energy, I know physics. I'm like, "Dang, dude, that's like an entire life knowledge that people acquire just for like that one subject and you have mastered all of these other ones. That's very impressive." Very impressive. But she came in for like 3 rounds, man. That was wild. But anyway, I thought that the discussion with the guy earlier was really good. I honestly that was like one of the best because I really feel like he really understood yeah, exactly, Gary. I don't try to say that I'm a medical doctor and people come in obviously because of my name or I am playing it up, right guys? Let's be honest. I am playing it up. But they come in and I don't try to say them anything but like a microbiology PhD. So yeah, exactly, Dr. T. Oh my gosh. It is like you do have imposter syndrome, right? Where it's like, dude, I have even for microbiology, I'm like, um yeah, yeah, thank you. I'm like, I do try to make it very clear. Like I'm not what you is, but this is what I was thinking, you know? Oh my god. Can you imagine a stethoscope? That would be so bad, dude. That would be so bad. I I agree. I agree, Gary. And that's the thing is that it's like, especially when science is being attacked as an institution and um, dismantled and not recognized, and anti-intellectualism and all that kind of stuff is happening. Like, there's no way that I'm gonna back down and let them like change and take titles away and take all that stuff. We can't. Oh, thank you. We can't let that. We can't let them take everything from us. That's just crazy. Exactly, Gary. Yeah, no. And honestly, when I see the physician, he like they love talking to me as a molecular biologist because they're interested in gene therapy and CRISPR and all that kind of stuff. So it's like they love talking to me about things, and it's cool, like nerding out about it, and they love it. And it's like the whole like it's all it's like that thing where, you know, professional athletes are all friends, but then the people that like don't play basketball and they're all like, you know, like Raiders or I die, you know, and like, oh no, I'm a San Francisco 49ers fan, and they get into like knife fights in the parking lot, and you're like, bro, the professional players are like hanging out and chilling, you know, afterwards, but like these fans are like fighting to the death. It's kind of like that with MDs and PhDs, right? Where it's like the perceived notion that there's this stigma against one or the other. It's like there really isn't. I've never experienced that in anything. Yeah, I think it's like actually kind of cool. Yeah, exactly, Gary. Yeah. Yeah, Dunning-Kruger's, that I'm always very afraid I'm on the wrong side of Dunning-Kruger. That is not a, you don't want to be on the wrong side of that graph. Yeah. Yeah, Nico, there is. I mean, there is like the whole like idea of if you're not working on humans, then you're, you know, you're somehow lesser than, or you have like the human mammalian molecular biologists that are like, well, my stuff is actually gonna like help people's lives, kind of thing. That does exist. So it is interesting, but it's fine. It's fine. You know, it's like it's inside baseball at this point, guys. Like we're all, those are so quaint times. I feel like those are just so hilarious, of like yesteryear, of like all those quibbles seem so pedantic now. Like we're like every science industry, both, I do both. Every science discipline is under attack. The entire institution of academia is under attack. Like there's no way. Oh, thanks, Gary. Thank you. I appreciate that. Um, Anti-intellectualism is taking us all out, you know? It's like there is no like, oh wow, no, we're gonna stay. Um, I don't mind that personally, Nico. I mean, but like we do like 5-minute rounds normally, just because that's what it is during tournaments. Yeah, for real, Gary. I think it's you know, I do love talking, you know, how to improve peer review and all that kind of stuff. Like we talked with that guy today, and I think that it's an interesting discussion, but at the same time, like, you know, we got to stick up for one another. There's a lot of things I can criticize about academia, but like I'm not gonna do that now. I'm not gonna do that publicly. I'm not gonna like, you know, make that seem like what I actually care about. I actually want like science as an institution to grow. You know, we have to like we have to band together. We have to support one another. Oh, thank you guys. Thank you. All right, guys. Well, that was fun. Thanks for chatting with me. I appreciate the meta conversation. You guys giving me feedback because I am new here, and I appreciate you telling me what's up. So I'm gonna do. Oh, thanks, Dr. T. Thank you. I appreciate you always being here. I'd love to know. I'd love to chat with you offline. You know, about your research and stuff, if that's possible or whatever. So, um, I'm trying to like do more data science. I tried to do more with it with my PhD research, and it didn't get to that stage. Kind of like got really pushed out because of the pandemic. So I had to kind of like do lab work, unfortunately. But yeah, I'm so interested in computer science in general. But um, thank you guys. I really appreciate you. So we'll do nightly streams, like 1 hour. We'll see how it goes. If I do like 1 hour streams, and you know, well, we'll have to see. I think with the timer, and if it fills up pretty quickly, then then it's good. But yeah, we'll just see. Let's just try it. Okay, 9 o'clock tomorrow, and we'll do it. So thank you guys. Thank you so much for being here. Thanks for being part of this community. I really appreciate you. Helping me out, helping me help you, help us build a better community. Oh man. Okay. I mean, that's so rough. I mean, that's like with epigenetics and methylation, we kind of like that's kind of we're coming back around, man. I don't know. I mean, Darwin at the time versus Lamarckian evolution that's a tough one, but it's kind of known now. Anyway, thank you so much. Thank you for everyone. Thank you for the gifts and everything. I really appreciate you. Thanks for the subs. Thank you, Jen. I appreciate you for believing and supporting the stream. Thank you for all the gifts. Thank you, Ale. Ale. How do I pronounce your name? Aye. Gary, Maiden, Dr. T, Jen, Hoyer, Ali. Hi, Ali. I appreciate you. I'll see you tomorrow, okay? 9 p.m. PST. We're gonna do it. We're gonna try nightly streams. Let's just check it out, okay? All right. See ya.